What if I told you that magic mushrooms didn't just evolve their magic, they borrowed it? Tonight, we're cracking open the secrets of psilocybin, the genetic mysteries behind it, and why mushroom names spark fierce debates in the mycology world. Joining me tonight is mycologist Edward Grand, PhD, here to help us unpack the science, stir up the controversies, and drop some surprises you won't see coming. Yo, welcome to the Myco Geeky Podcast, where we explore the fascinating world of mushrooms and the people who love them. From expert cultivators to passionate foragers, we bring you deep conversations, cutting-edge insights, and everything mycology. Whether you're a seasoned mycophile or just curious, we invite you to geek out with us on the wonders of fungi and join the mushroom movement. Hey, what's up, everybody? Welcome to the Myco Geeky Podcast, the show where loving mushrooms might be less about science and more about proving your friends wrong on a mushroom ID hike. I'm your host, Myco Geeky, here to help you dig deeper into the world of mushrooms and cultivation so you can take your at-home mushroom cultivation game from, like, nice pin set to, your mushrooms just unionized? Yeah. We got an epic show for you tonight, so grab your lab coat or your favorite hoodie and let's get growing. Tonight, we're diving into the evolution of psilocybin and the mysteries of fungal genetics with mycologist Edward Grand, PhD. We'll be breaking down how magic mushrooms may have borrowed their magic through horizontal gene transfer, explore what the oyster mushroom genome teaches us about fungal evolution, and settle the score on the old lumpers versus splitters debate. Does mushroom taxonomy actually matter for home cultivators? We're going to find out. Plus, we'll talk about how AFLP genetic testing might one day be able to level up our breeding game and confer successful monokaryon pairings. Get ready for some deep myco nerdiness. You won't want to miss this one. Before we jump in, please support the podcast on Patreon. It helps keep the show going and lets us bring you more deep dives into the world of fungi. And of course, hit subscribe, smash the like button, and let's get into it. All right, what's up, guys? We're about to do this. Ed Grand, PhD, in the house. What's up?
Man, this might be the nerdiest podcast ever.
Nerdsville.
I think we're gonna surpass like all the all of our talk about DNA sequencing and all this
yes,
nomenclature and everything. This is gonna be like way more nerdy.
I almost purposely put tape in between my glasses tonight just to set the perfect chef's kiss ambiance. Yes.
Yeah, I got some nerdy glasses, like real nerdy. I found one of my old pair of glasses from when I was like 16. They're like really, really nerdy. I understand why I didn't have a girlfriend until I was 21.
There's always a reason. That's all I'm gonna say.
Yes. There was many reasons. Yeah,
there's always a reason. Yes.
What is speaking of all this epigenetic stuff? Maybe generational trauma, or what do they call it? Yeah, transgenerational crises and trauma.
That was a lot of prefixes and suffixes.
Yeah, it's full of them. You know, what my mom ate or smelled or did when I was pregnant in utero, and all kinds of stuff that I can blame my mother for.
It's all her fault for sure. We already know that. We know that's true. Yes, totally true.
Yeah.
All right. So we, one thing that we like to do is just chit chat about what's going on in the community. And we have definitely had some interesting conversations. There are some people, saying some very interesting things, out there right now. And, we got to talking about it and decided that instead of getting into that so much tonight, we will get into it a little bit. But first, there were some papers. Ed, you had a paper. It was, just a sexy title. Molecular Karyotype of the White Rot Fungus Pleurotus ostreatus. Oh, baby. Yes.
Oh, yeah.
So I saw you put that up on your Facebook page, and I said, all right, I got to check this out. I read it. I read it before I thought to pump it through Notebook LM. My mistake, but that's okay. Let's start with this one because you posted it and seemed to suggest that maybe there was some implications or something of note worth reading. So tell me how you found this paper and what you're thinking about it.
I don't know. It started with I was looking into polyploids again, and so I was thinking about like yeah, just basically all these words, the aneuploids and polyploids, and polyploids are basically where you take your normal what you call a diploid genome or a dikaryotic genome in our fungi, and it basically you double it or triple it, and it has implications in all types of things like crops like wheat and maize and rice. They tend to be polyploids. And these polyploids, a lot of times they come from interspecific hybridization events. So that's where I started with it. And then again, it was like a rabbit hole. You just go down these things. So I was kind of basically trying to figure out how possibly in the future I could either make polyploid cubensis or pans and maybe even do like actual interspecific hybrids. So these terms have been floating around for a long time. Cube hybrids and mon-mons and dimons. Like once you get past that, you start to realize this whole thing is way more complicated. And what role like the mother. So in the sense of like a dimon, like a dikaryon mono pairing, the mon acts as the mother. So in the mother, like in humans, you get like a lot of the mitochondrial DNA. And then so in a dimon pairing, the monokaryon kind of acts like the mother. So I was fascinated in how some of these traits might be able to be forced into the new progeny by what monokaryon you use, which is again a idea that's been floating around for a long time. And then also the idea that maybe some of these, yeah, there's a lot of stuff to unpack, that maybe the conditions that we grow these things in might be altering the expression of the genes that are already there. And also this horizontal gene transfer idea that you could possibly pick up things from viruses or plasmids, or even, I hate to speculate, but even maybe other dikaryons. Similar in the way that bacteria, you probably are familiar with that, the way bacteria, they do this thing called conjugation, and there's these plasmids and these agrobacterium mediated transformation, and it goes on and on and on and on. So essentially I was looking for simpler ways to make like better and different cubes. That's kind of what it all goes back
all
to.
You're just trying to come up with another way to make more work for yourself. Yes,
pretty much. Yes, buy more random crap that I can fill up my house with.
Well, Ed, someone told me last week it's really easy to do a dimon, or sorry, not a dimon, a di cross. By that, guys, I mean you can cross a dikaryon with a dikaryon. You just put it on the plate and you just go, you dumb motherfucker, cross, you bitch. You just kind of get really aggressive about it and really angry, and they are so scared they just do it anyway. Yes, no.
Yeah, so that got me into asexual like reproduction and the transfers of these different DNAs and then the DDK thing, and then like when you do de-dikaryoticization, like are some of these like epigenetic factors, like methylated parts of the DNA sequences, are they being separated with the nuclei, and where are the mitochondria going, and what happens when you do these processes and you get rid of all the cytoplasmic elements and then you just isolate nuclei or protoplasts or spheroplasts or neohaplants or whatever the hell you want to call them.
Right.
So yeah, sorry, it's you guys. It's just like this. Which is why Notebook LM is really, really, really convenient.
It is nice. For those of you guys who have not gone to check this out, Ed plugged this, I think, a couple weeks ago. Notebook LM is free right now. You plug a PDF of a paper into it, and you can get a briefing summary. You can get study questions. And most interestingly, you can generate an artificially created AI podcast where it's two people talking through the gist of the paper. Not only that, now have you used the feature where you can actually give it advice? So if there's if there's an aspect of the paper you're most interested in, or for example, you're always reading these things to see how it can apply to what we do, you can actually tell it, focus on what implications this might have for at-home mushroom cultivators who are growing Psilocybe cubensis, I've done it. It will literally skew it. Now, what it tends to do, though,
Gosh.
it will pull it away pretty far from the paper and eventually turn it into this sort of general chat about cubes. But it was still interesting that you can tell it exactly how to make the podcast. I thought that was cool.
I think is that the interactive mode, the thing. Yeah, I'm seeing a button that says interactive mode beta. Yeah, there's a bunch of.
Yeah, and then you can tell it what to do. Yes.
Oh gosh, yeah. Well,
So
that's what's
there's
cool.
more.
It pulls in. It seems to also sort of be feeding on stuff that I've fed it before. I'm sure the AI, whatever
It
it
remembers.
seems to
Yes.
know kind of like what you wanna know already
Yep.
based on what you've been feeding it already. And yeah, it's pretty generic too, where it goes into like you can feed it some really nerdy paper, and it makes a lot of good analogies and kind of like metaphors. Like I've been writing them down, like this idea of like the genome as like a cookbook, and like certain genes in that cookbook being like the recipes in that cookbook, and like the expression of those genes, like the actual cook who's making the recipe, and it's some really really nice analogies for every so we don't have to talk about dogs all the time.
Yes. I agree.
Dogs.
Yes. I can't remember which paper I did not too long ago, but there was a concept that I sort of understood, but yeah, the really cheesy, hokey podcast host had a really slick way of describing it. And I was like, oh, damn you, AI.
No, well,
Yes.
my friend, I played one of one of my friends, and he was like, "Oh, she sounds sexy."
Yeah.
So the female host on the little podcast it generates also, if you need some like bedtime stories about
Yes.
amplified fragment length polymorphism.
That's what I'm looking for. That will not put me to sleep always, though.
I
I
know.
can't wait. Just think, we're not that far away, right? You're gonna get the free version where you don't get to pick the host, but for like another $10 a month, you're gonna get to have like Snoop Dogg talking with, James Earl Jones about AFLP and its implications of
I'm sure,
exactly.
like the LBC ghetto version of Yo
There,
Mammy.
there you go. There you go. Yes,
I don't even want to know what kind of analogies it'll come up. Then,
bro.
like,
I
yo,
mean,
when you walk into that liquor store, you got 40 some
yes,
malt, you got OD, you got Coke 45.
yes. Yeah, man, you might get some really colorful metaphors using a different character. You never know.
Yeah.
Yes,
That'd be
lot of implications. A lot of implications for sure. So, so, all right. So, what did you ultimately figure out from that paper? Like, because what I was figuring out
that
is,
I need to go back to school.
Yeah. Yes.
I figured out that I'm really stupid. Like I'm way more ignorant than the old Dunning-Kruger effect. It's like I'm way, way behind.
Yeah.
And
Well,
these are like 20-year-old papers. So like,
right.
oh my
They're not,
gosh.
the couple that we're talking about are not brand new papers. Now, I thought it was interesting just that it didn't seem terribly complicated to section out the chromosomes,
Yeah.
which I thought was cool. And so the idea is you might, if you could do that for different species, it would give you some clues into mating behavior. Is that right? Am I thinking about that correctly?
Yeah, I think there would be one application if you could find, so they call them chromosome length polymorphisms or
Yeah.
CLPs. And I remember it's funny because I remember one of my co-advisors mentioning this back because I started my PhD in 2000, and I remember her mentioning this paper because they've done it also in other organisms like corn, like maize. And I don't know if you remember this lady Barbara McClintock. I think she ended up getting a Nobel Prize. Oh God, it started raining here, so if I get the power blips, you guys, it should be
Okay.
okay. But it hasn't rained for 3 months, and now it's like raining.
Okay.
But yeah, it'll get rid of some of the smog and the air pollution, the rice smoke. But yeah, it goes way back. They had studied this woman noticed at one point a long time ago that chromosomes, which you can actually see under the microscope with the proper staining, they're fairly easy to see. And so you notice that there was these abnormalities on some of the chromosomes. Some of them were kind of bent, and this was kind of way before DNA sequence and your whole genome sequences, WGS. I mean, this was just literal like physical changes in the chromosomes. Short. Probably everybody's seen on the wall of some biology classroom. The 23 pairs of human chromosomes, and you've got this one, number 11, number 12, number 13, and so this is what you call karyotyping. Which karyo, again, back to like monokaryon, dikaryon. The karyon, it refers to the nucleus, and in particular in this way, the chromosomes. And so this is like kind of a more sophisticated way. They use this, what's called pulse field gel electrophoresis. And they basically could separate the chromosomes without denaturing them. So it's kind of like a Lego blocks, and you would take the different size Lego blocks and you can separate them, and just straight away you can tell how many chromosomes are in the organism. So
And
the
what was that
haploid
using electricity or how was it doing
yeah,
that? Yeah, okay.
it's gel electrophoresis.
Okay,
And so
so it's using that same, I got it. But it's a
yeah,
little different than like for PCR.
yeah, well, so it would be the products you check after the PCR reaction.
Okay.
So when you see those like gels, the polyacrylamide or the agarose gels, and you see like the little ladder thing,
Right.
those would represent, instead of pieces of DNA, which would be your amplicons from your PCR, they would be actual chromosomes. And so
Okay.
based on how quickly they migrate in that electrophoretic gel, it just basically uses electricity, and it's like the smaller fragments move faster. So if you had a smaller chromosome, it would just move faster down the gel.
Right.
And so based on a comparison to a reference, which is actually called the ladder, you would put some kind of size standards in there, and you could tell how large those chromosomes or piece of DNA are. And so if you were compare, say, like an oyster mushroom to a cube or two cubes to each other, you could tell the difference. Almost like a fingerprint. I hate to use that word because it gets used for several different things, but
Right.
almost like a barcode or a fingerprint. But in this case, it would be for the particular chromosomes that were in that organism. But you can do the same thing with, like, I don't know, like proteins and DNA fragments and
Right.
actual DNA. That's usually what people use the term barcode for. Um,
So
so
but
yeah,
they're not doing a PCR process beforehand.
no,
How are they breaking them up then? How are they getting
ba-
the
basic
yeah,
sonication. I think I don't
sonication. Okay.
recall a lot. Yeah, lysis buffers and sonication. So you would just say, I don't remember what exactly they did in that particular paper. You just use things like soap, basically, like SDS and polyethylene glycol and things like that to just dissolve the cell membrane. So you just end
Okay.
up getting a solution, kind of like the way you would extract DNA. Very, very similar to that, except you try not to break it up. So a little bit gentler process probably than you would use for extracting DNA. So you just kind of, you break open the cells, you lyse the cells, and then you've got all these chromosomes floating around in there.
Right.
So yeah, pretty straightforward
So now
thing.
this is interesting in the sense for me that it reminds me of this AFLP thing because at the end of the day, if somehow some way we can get the right primers, all we're doing is run to do our little paternity test. You're doing the same thing. You're breaking it all up by these, finding the right places to grab the
Hmm.
unique parts of the genome for your two parent fruits and your so putative offspring fruit, and then you're just running these gels, and the lines should all line up with the lines of the kid should line up with the lines of the parent is the idea.
Yeah.
So it's still getting me, I feel like every little paper you're sending me or you're posting to Facebook is really just keeping the dream alive for me about this
Oh,
AFLP.
yeah. Well, so the main difference in all these techniques is the level of resolution. So let like start say if you had like 10 random humans and you did karyotyping of their chromosomes, you would see that they all have 23 chromosomes in the haploid. So usually you deal with the haploid like a monokaryon. You only want half the genetic material because it just makes things simpler, especially if you're trying to, do things like unravel like where the parents come from. It's
Right.
usually easier if you work with haploid genomes. So if you basically if you wanted to see if like everybody was a human, you would say, oh, well, they all have 23 sets of chromosomes or 46. Well, that doesn't really give you much information. But then if you're talking about, well, maybe you want to know, like, are these particular, maybe people like this family in this family, they're related. That might be something where you would use maybe something like AFLPs or even maybe like a paternity test. You would look at like a not chromosome level, which is, chromosomes are big, so you go from this like in cubes, you've got like 46 megabases, and then in each one of those chromosomes, maybe you've got a chromosome that's 6 megabases, and then with AFLPs, you just kind of go down to like, I guess, higher resolution. You can look at finer structures. So instead of like population levels, you go to like family levels, and then maybe like individual levels, and then even in that individual, maybe where their actual, so you could actually do like family lineages and stuff like that. And then finally, you get down to DNA sequence. And then you can talk about the expression. So some of these techniques, they look more at the expression, and that's a whole other different thing. But there's these different levels of resolution, and sometimes they give you more or less information, and it kind of depends on what you're looking for. It's really it's a big huge topic. But as far as like just comparing two individuals and seeing if they're related or what their parental lineage is, yeah, there's at least probably 15 techniques. There's ISSRs and STRs and RFLPs and AFLPs, and the acronyms just go on and on and on. And so they all have their own kind of uses depending on what exactly and what your lab is equipped to deal with. I mean, as you know, like, if you got say primers for AFLPs or your adapters and all these things, and you say somebody's working with some plants or whatever, maybe sunflowers, and you want to work with roses, and somebody else wants to work with like orchids or something. If you're already kitted up to do one particular technique, new graduate students or new researchers will probably just continue on with that technique. So they almost become like kind of, they become sort of, I don't know, like grandfathered into certain labs. So you see some labs, oftentimes they'll put out papers like using the same technique, but maybe like different groups. So if you're working with oyster mushrooms, it would be really easy to apply that to shiitakes or cubes. So.
Right. All right. So I think so far in my research, I am secretly hoping that one of these days I can find somebody that's already done some of this work in a PhD level program or postdoc or whatever, and this is kind of their jam. And fingers crossed that maybe we can get lucky and out of the gate not have to buy too many of these restriction enzymes and all that. But it is entirely possible, and based on the quotes I have been getting from these private labs, their quote is definitely not assuming this is going to be an easy process. So we'll see. But it is potentially the case that you would need a different set of different couple restriction enzymes for different goals as well, right? Because this is what you're talking about, resolution. That you would need a certain set to get the resolution you need to test for paternity, whereas you would also need a different resolution to test within the same species whether there's a discrete difference between, I think I've said this before, a zap growing on one side of a riverbank versus a zap growing on another side or whatever. So even though geographically they're close, how do you really tell if it's the same or different?
Yes.
You really don't know. I was just talking to somebody else recently. I'm not going to name names, but he and I were talking about this idea. And he said, oh, it's interesting. I was just talking to so-and-so who, I'm not going to say who again, also super, super famous, real, legitimate PhD holding mycologist such as yourself. And they said that they were told that if it was within 10 feet, that it was definitely the same, just definitely the same thing. and how they were like, well, okay, that probably is a fair thing to say, but do you really ever know? And because, I mean, look, I've seen people who have harvested pancyans from their yard, and then they show a photo of it, and some really observant person on the internet goes, you see these three right here, those three, those are actually Phonsekiais, and you better not eat those because those are going to make you sick. So,
Yes.
like, how do you really know?
That was the classic Gallerinas mixed in with these Cooneromyces and other
Sure.
Foliotas and things like this, or
Yes.
even like a Flammulina. And then, yeah, you see them at like Namaphore, they're just all on one pile, and somebody comes along and is like, there's a Gallerina in there. Like, that's
Yep.
not that's not a Nematoloma or a Hypholoma. Like, why there's a Gallerina, and like three of those will kill you. So
Yep.
it's like, whoa, yeah. So,
Yes.
but yeah, that's a little bit different than this population level studies. But yeah, you're right. Even within one log, like we've talked a few times about the armillarias, in one log you can get these what they call intersterility groups, where you have it appears to be individual diploid or dikaryotic. Armillaria, as we've talked, is like one of those weird basidiomycetes where it can exist as a diploid in the vegetative state, but mostly it's dicaryons. But yeah, people were doing that research back in the 80s and 90s.
Right.
The forestry people, they're like, these armillaria that are pathogenic. They're like looking at these logs, and this started, I don't know if you weren't really around then, but back in the day it was armillaria. Everything was just armillaria. It was Armillaria mellia, and that's it. It was honey mushrooms. And then they started off with this North American biological species, the nabs, and you had nab 1, nabs 2, nabs 3, and now we call them gallicle and astoi. And you probably are familiar with, I think in Ohio you guys get the astoi, astoi, armillaria. And so what you would call a honey mushroom in Ohio is not the same as like Tennessee or South Carolina or Florida. And it definitely not out in Oregon. Like these are all, so I think they came up with 9 different biological species. And the reason why is because they were morphologically, there's not a lot going on there. They pretty much look like honey mushrooms. Of course, when somebody tells you they're different, you're like, oh yeah, no, that's obvious. Like, yeah, that's obvious, like Panulus phoenicia, and that's a Cinctillus, and that's whatever. But until somebody tells you, you're like, oh, they kind of look the same and they go on the same pile. And
Right.
then when somebody actually separates them, you're like, oh, now I see. Okay, yeah, that makes sense. But to the average person, like collecting honey mushrooms to eat, it doesn't really matter because they're all edible. So you're just like, yeah, they're honey mushrooms. Go
Yep,
fry
but
them in a pan.
like they say, you don't get a PhD eating them.
Yeah.
You don't get a PhD eating them. You got to do
Said
something
that?
more. You got to do something else. Yes.
Oh,
Yes, yes, yes.
that's. I
Yes.
want to put that on a shirt.
I
Oh,
like it.
speaking of that, like I was going through my closet and I found this isn't old. This is the University of Tennessee fungi. I don't know if you can
Ooh,
see
I
it.
like it, dude.
Yeah,
Y-
these were made. This was way in the back of my closet. I was digging out my closet because I'm getting ready to
Nice.
move, and I
God,
found
did
this.
you weigh like 30 pounds more in college? Because that looks like a
Yeah,
big shirt for you.
well, these were some of the last ones.
Oh,
This was
okay.
back when they had to print. You had to print like 200 shirts at a time.
Right. Oh
And
yeah,
so I
back yeah, not on demand. Yes.
Yeah, these were, and very, very simple graphics. This is probably
Yeah.
like it was highly coveted. We would wear these to the conferences, the international conferences, and
Right.
be like, like a little gang, a clique.
Yeah.
You could always tell Ron's like former students because they'd all be wearing these shirts. It's like represent
Love it.
UT. I don't know,
Love it.
IG,
Love it. Love
UT,
it.
something like that.
So did you guys have a did you have your own walk? Like the Crips got a Crip
No,
walk. Did you guys have your own
I
Tennessee
don't
walk?
I don't think so. I don't think so. We
Okay.
just go out and get drunk together and then like bitch about Ron and what a dick he was.
Yeah. 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 Yeah.
Like, oh
Shout out
yeah.
to Ronald Peterson if you're listening.
Yeah, if he's out
Yeah.
there, he's probably stay. I'm sure I'll catch one of these one day. Props
That
to Ron, my second dad.
that would be hilarious. That would be hilarious.
Representing.
Yes.
I need to get better at gang. I only know the blood one.
Did you just say I got to get better at gang signs?
Yeah, yeah. I'll
Oh
put
my
ask
gosh.
AI to make a UT gang sign.
That is funny. That
The
is funny.
Vols probably something with a V and a U and a T or something.
Yeah, just figure it out and roll with it, and you did it. I mean, no one's done it, so if you do it, you win. It's yours. It's yours for the
So,
taking.
I don't know, 'cause
Yeah.
I'm so ghetto.
Well,
Knoxville, Tennessee, man.
I think that's sort of like the right spot to move into. We were gonna talk more generally about taxonomy. What is it? Why does it exist?
Ooh.
Because your PhD specifically was couched very much within the concept of it, because your thesis was trying to fix some of it. Your
Mhm.
buddy Mr. Peterson was definitely that was his jam. So
Mhm.
so yeah, maybe just start us off real simple for everybody watching who hasn't heard some of our previous talks. Just generally speaking, what does taxonomy mean? What does it mean within the context of fungi and mushrooms? And then we'll go from there.
Yeah, that's a great question. Nobody's ever asked it in that way. And I think it's quite simple. Taxonomy is the way we separate things, and nomenclature is the way we give those things a name. So the underlying idea behind taxonomy and nomenclature is to give things names so that we can adequately communicate with each other. So when we have, this is one of the things, like you've got a barcode, a DNA barcode, right? But maybe it's 800 base pairs. You can't say like, well, yesterday I was out in the woods and I found a session number 43296.1, and that's got an iNaturalist number of PD4KHT5. But you need, so you can, but if I say, oh, I found Lactarius volimus, like then you know what that is, right? So I need to communicate those things, and even though the Latin's a little bit tricky, you get used to it after a while. Like anybody out there listening knows what Paniola senescens is, right? But without, you have to have a name for these things. You have to have some tag to, communicate well with each other. So the difference, so taxonomy is like, let's say you've got cars, and then you've got trucks and cars, and then you've got like sedans, and you've got station wagons, and then you've got sedans with like 2 doors and sedans with 4 doors. So that's where the taxonomy, like, am I going to call a sedan with 2 doors a different thing than a sedan with 4 doors, or am I just going to throw them all together and call them sedans? So that's what, like, the lumpers and the splitters, where that comes in. And I'm going to call all sedans like Toyotas and Mitsubishis and Hondas. Like, do I call them all just sedans and just like kind of create this artificial group? So taxonomy is a lot about artificial separations between groups that particular person thinks are worthwhile, or they're it's very subjective. So to me, if I saw a Toyota, a 4 door Toyota and a 4 door like Mitsubishi, I might not know the difference. But to somebody who studies cars, they would be like, Ed, you dumbass, that's a Toyota, that's a Mitsubishi. But to me, as a non-car person, I just see a 4 door sedan. I don't know. Like, that to me is as good as it needs to get. I know it's got 4 doors. I got to take the kids to school. That's good enough. But to somebody who studies cars, that would be like blasphemous. They'd be like, "Oh my God, Ed, I can't believe you called that Mitsubishi a Toyota."
Or the other way around, or probably, yeah.
Yeah,
I feel like it might be the other way around. But that's it.
I,
Even that metaphor is interesting, because depending upon the person, they might lump cars together for different reasons. One person might look and go, well, all the 4 doors are one kind of vehicle, and then all the 2 doors are one kind of vehicle. Another person might go by color, and another person might go by sedan versus coupe versus hatchback. So even from the perspective of
exactly.
who you are and what your concern for the thing is could change, which I'm bringing
Mhm.
up because even within the scientific community, there are different ways to be a scientist. Even as a mycologist, there's different ways to be a mycologist. You could be a genomicist. You could study
Mhm.
just ecological factors. You could just
Yeah.
study pathogenic properties of the mushroom or fungus. And depending upon what your actual area of concern is, that might change your priorities
Mhm.
taxonomically and nomenclaturally, right? Like you
Mhm.
might go, well, for my research, I need to separate them all out. But honestly, for a lot of ecologists or a lot of people from, mating and commercial interests, it might make a whole lot of sense to lump things together, right? Like we have certain clades, we have certain
Totally.
things where you're like, okay, cool, we proved it's something slightly different genetically, but functionally speaking, that only matters for the people who are making a big deal about it.
Yeah, I mean, yeah, you could go back to the car thing, but maybe the fungi thing, like colors in cars. Maybe I live in hot ass Thailand. I want a white car. I don't
Right.
care what brand it is. I don't care. I just want a white car because I want it to be cool or relatively cool in the daytime. So that might be very important. If you're an ecologist, you might just be interested in what are the wood rotting fungi, or maybe even what are causing brown rot, or what grow on conifers, or what grow on oak trees, or hardwoods, or softwoods. And you can make these delineations based on, like you said, whatever is your particular interest. It could be something more like particular, like insect pathogens. Maybe you don't, I mean, there's a lot of fungi that grow on insects. It could be just something like basidiomycetes and ascomycetes. And yeah, you can chop the whole kingdom of fungi up in so many different ways. And that traditionally has been done with microscopic characteristics and a little bit about their ecology. Like if you get into wood rot, wood lovers, not wood lovers in the sense we use it, but I mean just like fungi that grow on wood,
Right.
like Gymnopolis and whatever, Armillaria and things like that. You'll see that they immediately separate them into the brown rot and the white rot fungi. And so that could be, tricky too, because what if you have a certain genus of mushrooms that have been traditionally grouped by their microscopic morphology, and then all of a sudden you realize that some of them cause brown rot and some cause white rot, and what you think was a significant taxonomic like way to separate them becomes not so significant. And this is where that horizontal gene transfer and all that stuff comes in. But in my genus Lentinus, for instance, there was one species, Neolentinus lepidius, that was separated because all the other members of Lentinus were white rot fungi. And this one particular species, it grows on like it's called a train wrecker, I think. It gets to be quite large, and it used to grow on railroad ties. And they're like this is obviously a different type of fungus. And so Redhead and Scott Gins, I think is his name, Scott Redhead and somebody else. Anyway, they separate it into this separate genus, and at first they got a lot of flack for it because it was just this one characteristic. There was actually a couple other characteristics. And when they first separated into a new genus, they split it from Lentinus. Everybody gave them a ton of shit for it. And part of my job during my PhD was to make sure they were right or maybe wrong. It's always better if you can prove somebody wrong as a graduate student.
Right.
And so I get a
It's
feeling
a better flex, right? Like it's a way better
yes.
flex. Yeah.
You don't want to confirm what people already know. You want to create some controversy
Yeah.
and be
You're
like,
not gonna
look,
get a
that person's
yeah.
wrong.
Yeah, you don't get a PhD going, yes, they're right.
No, exactly.
You get a PhD going, hey, Mr. Scientist, I'm a new Mr. Scientist. Yeah, and I figured out why you're wrong. Okay, so what did you find out with that one? Like what from the barcoding information? Did it want to be in with other Lentinus?
No,
Or
they were right.
They were
They
right.
were
Okay.
right. So
Okay.
so I didn't pursue it any further.
Nice.
I look for other people to prove wrong.
Yeah. Man, I'll tell you this though. There are some genera. I mean, I'm just doing some of this sequence validation with fundus. Just enough to start getting a taste that sometimes it's super easy. Sometimes it just there's enough evidence. It all makes sense. And then there are other genera. Certain genus are just naturally a fucking hot mess, and it's really hard. So that's why I was interested in what you found. Did you find, oh yeah, nothing about this ITS is really jiving with the rest of these, they got it wrong? But now what do you do in a situation where it looks exactly the same and yet the ITS says put it somewhere else? How,
Yeah,
like, has that happened? Do you recall any instances? And
yes.
like, as a lumper, what are your thoughts about that?
So 2 days ago, this happened. Scott Astuni sent me a message, and he was asking me about this picture on iNaturalist, and I'm like, "Oh, that's Bateriae." And I would have, I so I didn't actually see the sequences, but I was kind of torn. It looked like what I would call Bateriae, which is related to crinitis, which is one of the ones I studied. And it was one of those things. It wasn't fitting in the tree where it should, and I don't know what he. I think that's what he ended up calling it, just for the time being.
Yeah.
So it's another one of those kind of sampling things. If you don't have a lot of samples, if you just have a single collection, it makes things a little bit more difficult because you just need to put a name. I mean, there's a certain like practical aspect to these things where you have to move on, and
Yeah.
it's a little bit frustrating. I think I heard some quote the other day, like when you a lot of these papers or your PhD, they're finished when you not when you're happy with the results or you think you're done. It's when you learn to hate it.
Yeah.
Like that's when it's done. Like you have to
Yes.
just
Yes.
like
Yes.
get
Yes.
it over with and move on because you'll
Right.
never be 100% right. There's always this like feeling that that is not as good as it could have been, but you just have to move on.
Yeah.
And somebody else will correct you later or later. Some like, smart
Yes.
ass like or AI will come and correct you later.
Exactly. Exactly. Yeah,
So the
we get some of them where you're like, well, because when I was first starting to learn this, I was like, so what do I put in the ones where it's not where I really don't feel confident saying it's definitely this genus and species? What do you say? And they were just like, well, you say what you figured out and what you don't know, and that's where you leave it. And then someone else can, the goal is hopefully you did something that was somewhat helpful to somebody at another point in time.
mm-hmm.
But yeah, the it's like that with people, right? In types, there are burnouts, there are goth girls, there are chess nerds, there are jocks, there are right, there are all these types, and then every once in a while you meet somebody and you're like, he don't fit. He doesn't really fit in one of these slots that people usually fit in, right? There's definitely anomalies. So I guess fungus can do the same thing.
Yeah,
Sometimes,
yeah. They're doing
sometimes
a
they
lot
do.
of that. My friends recently have been watching way too many of these like self-help podcasts and things about like childhood traumas. And I'm like, I should stop like analyzing people and becoming like an amateur psychiatrist or psychologist or whatever. But some of it's fascinating, to
Yeah,
like
yeah.
it's the taxonomy of people. And you're just like, I wonder what's wrong with that person. What
Right.
was I do it all the time. My buddy, he's from South Africa. He is all fucked up. He's like doesn't know he's like a case study in like psychological problems. Sorry,
Yeah.
Bernard, but if he listens to this, he's like, we try to figure him out because we're sitting there, we'll talk, and he's like, my parents are this and that, and I don't know,
Yeah.
like the generational trauma and different like heritage and countries and families moving, and yeah, you have all these overlapping influences, and it's, I don't know, my life was pretty simple, I think. I just like had like divorced parents that like used to argue all the time, all that stuff, normal stuff, but
Just a good old fashioned divorced
yeah,
household.
good
Just
old good
run of
old
the mill.
American
Yeah.
marriage,
Yes,
mom
exactly.
moving out of the house, dad moving out of the house,
Yeah.
broken plates, things like that.
Broken plates. I remember those days too. My parents are still married, but I do remember a couple broken plates a couple
Ugh,
times. Yes.
yeah.
Yeah, man. I think sometimes, so there's, of course, there's the real cut and dry, you can have schizophrenia, you can have bipolar disorder, you can
Mm,
have personality disorders, and they're pretty cut and dry. But when they get real complicated, and I've taken care of these people, is when they got a few of those things. There are literally bipolar schizophrenics who also have borderline personality disorder, who also have narcissistic personality. And just there's a point where you're like, bro, I get the psychiatrist is just trying to identify it all and figure out what to do and document everything. But sometimes it is just good old fashioned, whatever it is ain't working. Yes.
mhm.
It doesn't always need a name. It can just be Yeah, somebody's maladjusted, somebody's not fitting a certain mold. And ironically enough, there are plenty of those people in our mushroom loving community here.
Oh yeah.
We
Yeah, have you
we
seen the
encounter a few.
DSM recently? The book that there's it's
Oh
online now,
yeah,
of
it's
course, but
huge.
man,
Yeah.
the DSM is just like crazy.
Yes.
What is it? The diagnostic something
Something manual.
manual?
Yeah.
Yeah, the DSM-5. I think it's like
Yeah.
5.5
Isn't it?
seven
Oh, I
now
thought
or
it was like
something.
I thought it was almost on 6 now. Oh, maybe.
Yeah, I think they were making dashes. I don't know. I looked it up and then I started reading some of the entries. I'm like, oh my gosh, it's like the I whatever they call it, the ICBN, the
Yeah.
the Botanical Nomenclature Guide. They got another name for it now too. It's like thousands and thousands of pages. It's like the legal document for how to do nomenclature. So if you want to do nomenclature, if you're really, really super nerdy, you can go look up this thing. It's called the International Code of Botanical Nomenclature, but they've included fungi and bacteria. Like we think we've got it difficult with fungi. The bacteria people have it way worse because
Do they?
bacteria don't have any, yeah, they don't have any morphological characteristics.
Oh,
They're
right,
just
right. They have it
they're just
difficult in
like
that way.
rod-shaped bacteria. Like, how do you differentiate, lactobacillus from, I don't know, streptococcus? So, well, there we the
Right.
shape, right? But once you get past that, they're either a rod-shaped or
Or
they're
a
helical or
cone.
they're,
Yeah. Right.
yeah, like beyond that, there's like 1,000 kinds of like rod-shaped bacteria you could find like growing on your mobile phone,
Right.
like on a doorknob, and it's just like, yeah. So now it's called the
But
International Code of Nomenclature for Algae, Fungi, and Plants. It's got a
right.
website, and it's
And your thesis advisor at one time, he oversaw like a major revision of that.
oh yeah, he was one of the
Exciting.
like editors of the book form when it was. I think the last time they printed the book form, it was about that thick. Was like maybe 2004, and then it went online after that. So yeah, I think they used to call it the IMC International Mycology Conference, and they would have a little breakout group that did all the nomenclatural stuff. Yeah, and he was like one of the editors, chief or whatever the hell you call him back then. He probably still does it, but yeah, if you wanted to talk to a real nerd about nomenclature, you could get in talk with Ron Peterson, and you would yeah, he would talk to you for hours and hours and hours about the intricacies of why Flammulina is not the proper name for velvet foot like Enochi's.
Right.
Go
Yeah,
back
that
to like
those are
1786 publication by
yes.
Fries and this journal in German and that crazy.
But that's about to get way easier because of AI. You just once there's like a mycology edition engine for AI, you'll
Mhm.
the ICBN can pump in all their rules and it can easily tell you like yes your name for your new winter teacher works or it doesn't work and
Yeah.
here's why and yeah all
Well,
that should
it's
get easier.
kind of like the legal system where we have clear-cut laws, but at the end of the day, there's a judge and a
Right.
jury. So even if you have all the facts, you still have a judge and a jury. And so even though there's the rules for nomenclature, there's exceptions. Like let's say people have been using the genus Armillaria for the last 100 years, and some smartass or AI comes along and says, well, in 1769 there was a publication, but before freeze, and maybe we should use this. There's judgment calls where you have to do at some point what's also practical. So if every mushroom book, every mushroom ID book for the last 50 years has been calling it armillaria, maybe we should just keep calling it armillaria,
Right.
even though that's not the right name. So there's I guess like protocols or things for like what they call conservation. So that's what Ron was the head of this committee that basically comes together and everybody's like, well, it's not the right name and we know it's not the right name, but maybe we can conserve it. And maybe we can just like let this one slide. Everybody knows what it is. Let's just continue to call it armillaria.
Oh,
Like we know it's not the right thing. It's kind of like if you convicted somebody who was like mentally handicapped of a crime that you would normally convict a fully capable person of. You know, like if somebody steals a candy bar, right? If they're like mentally whatever the PC word is, how you know what I'm saying?
MRDD is what we say. Yeah.
Yeah, MRDD. Like you're not gonna throw that person in jail for 3 fucking years because they
Right.
stole a candy bar, right? But if it's an 18-year-old who maybe is fully,
Yeah.
so there's rules, but there's kind of like you gotta sometimes exception. There's
Right.
exceptions. There's a
Yes.
practical aspect to it.
Yeah. I get
Talk
that.
to
That makes
anybody who's been through
sense.
divorce; they'll know all about it.
Oh, will they ever? Yes. Yeah.
A
Yes.
split
Yes. Yes.
50-50.
Yeah.
I've been talking to too many
Oh
disgruntled old
man.
white dudes who are not
Nope.
happy.
Nope. 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 Definitely not.
It's always,
I talked to your I talked to your buddy Juan Luis
oh yeah, I
at
just emailed him
at
yesterday.
Alabama, and I sent because of you were like, well, I'm in Thailand, I can't send you any genetics, but reach out to my buddy Michael Geeky. So I sent him some. His student did a cool paper, and I guess now the university seems to be warming up to it, and he's got more people
Oh,
wanting to do projects. So we're gonna get them more stuff, and they're really hoping to figure a bunch of stuff out about media and protocols for media and all that kind of stuff. So we'll
mhm.
see how that all works out.
Oh, that's
Um,
cool. I
but
I've been playing with media a lot too recently.
it's fun. It's fun.
Yeah, it's like it's pretty simple stuff, you can kind of just do at home.
Yeah.
Start boil some carrots and use like carrots for your agar instead of like potatoes.
It's the leftover water. I'm like,
Mhm.
yeah, leftover water from rice, leftover water from potatoes, leftover water from insert whatever you want to do.
Mhm.
You know, but the minute you start doing that, you're gonna have some guy going, "Oh, you gotta buy my plates. It's new Skittle water, bro. It's
There you
Skittle
go.
water, bro. Skittle water is the best mycelial growth. I'm telling you." Yeah,
That's what people.
but who knows?
Yes,
Maybe it does. Yeah,
could
yeah.
be. Could be secret ingredients, man. Shh, don't tell anybody. That's my new project.
Yeah, well, the
I'm gonna
secret
have to give it an
ingredient
acronym.
can just be that you didn't put a secret ingredient in, and
Yeah,
you at least get to say it on there.
or you just
Yeah,
repackage some bulk stuff that you bought online.
there you go. Oh, so
Yeah.
I was just talking to somebody about this, and he's talking about how pre-sterilized grain is so cheap. He's like, this shit's $3 a pound, and when I'm buying my popcorn's about $1 a pound. So
Mhm.
like I think I get it for 87 cents, but let's say about $1 a pound. So basically, uh, for $2 a pound, somebody else can do all the work for me. And I was like,
Mhm.
yeah, it's starting to get pretty cheap. And if you buy in bulk, if you buy like a pallet from somebody, they give you an even better deal. So it starts getting pretty cost effective to buy the stuff.
Mhm.
And he was saying, I wonder how many people are working out deals with these major grain and sub suppliers, and they're getting their order shipped, and then they just throw it in their
Mhm.
fancy box and just sell it. I'm like, I bet there's plenty of people who do that. You never know. So now you got two
Oh
middlemen,
yeah.
and that's
Oh
where all your
yeah.
problems happen. Yeah.
I was the same here. I didn't realize there's like one I thought was just a little mushroom farm down the road, but they make the blocks. They make they grow in these like kind of bag blocks, and
Right.
you see them everywhere in Thailand. And I thought that like everybody made them themselves. No, it turns out this like little farm that's got like a one of those like atmospheric sterilization chamber things. It's like a container, and they make like all the blocks for all the farmers in Thailand. It's the same thing. The
Yeah.
Shiitake people, they get their blocks. Like only small farms now make their own blocks or their own grain. And once you invest that $4,000 into a autoclave, you got to use that thing.
Yeah, you do.
Nobody needs 150 liters of grain per week. So you're like, I got to run the thing. I got to turn it
Yeah.
on. It's like, yeah, that's what are they commodities of scale kind of thing.
Right. Yeah, I was about to go into metallurgical sciences as a kid. I got a little not a fellowship, whatever it was. So it's like a smart, nerdy kid that got to go hang out at this castings plant in West Michigan called, at the time it was called Howmet. I think now it's owned by Thiokol. But anyway, I was there. I figured a couple things out for them while I was there, and they were really begging me to stay. They didn't want me to leave. And I was like, I want to go get a degree. They offered me a little bit more money. I'm like, nah, that's really nice. It is very tempting, but I mean, I want to go get a fucking degree and really learn how to be an engineer. So they're like, cool. And then I had to take a meeting before I left with what I thought was going to be one attorney and get me to just sign some little non-disclosure. It was a room, and I swear to God, it was 7 fucking guys in a room. I'm assuming they were all attorneys. They had 80-some page non-disclosure, and these guys were not even trying to be nice. I'm a fucking kid who got like a, come see what it's like working here kind of a gig in R&D, and I don't think they expected me to figure anything out. I convinced some guy to give me a PO and start doing some research, and figured something out for them that even one of their in-house guys didn't figure out. And so they absolutely 100% were like, you should have taken that job. It's still on the table. Are you sure you don't want to take it? If you go get a degree in engineering and you try to work in engineering, you better come back here and work for us. And if you don't, we're going to keep our fucking eye on you. And you cannot ever do anything related to what that research project was, or we will sue the living fuck out of you, and you will never work in engineering. I'm not lying to you. This is literally what they did. They scared the shit out of me, to the point where I was like, fuck engineering. I don't want to fucking work in a place like this. This is what they do to a kid just because he figured some shit out. I ran into my boss, like, I don't know, 18 years later at a music festival in the summer. We get to talking, and I'm like, did you guys ever do anything with that research project I figured out? And they're like, oh yeah, that's how we do it now. Yeah, it basically, this guy Rick, who is their PhD metallurgical scientist, he ran with your research, and we got it all dialed in, and yeah, it works great. And I'm like, oh cool, I have to go look up my name on that patent. And he goes, yeah, your name's not on that patent. I said, that was the one fucking thing I asked them to do, was make sure my name gets on that patent. Yeah, well, that was never going to happen. And I'm just sitting there going, I made the right decision. I don't want to go, help a big company get super rich, never get any fucking credit for it. Yeah. But those blades, I figured out basically, if things worked out the way I thought it probably was going to work out, they were selling these blades to the U.S. government, U.S. military, for about $78K a piece. And it was at the time costing us about $56,000 to make them. We were taking 2 slabs of Ti-6-4 titanium alloy. We were milling out the cooling channels. We were bond coating them, heat treating them, and then doing like the metal X-rays to make sure they came together exactly perfectly. And then we'd heat it up and twist it, and then they would fail at that point, and then we'd mill it down. It was like a whole rigmarole. And all my idea was, well, why can't we cast this thing? You can't cast it because of the cooling channels. Well, what if you used a really soft steel? You could leach it out later. Oh, no, we looked into that. I can't do it. Well, I figured out how you could do it, just using different molarity baths and all that. And so now they're doing it. I'm guessing they're making those for about 18 to 20 grand a piece now. So saving them a lot of money per blade. I probably made that company hundreds and hundreds and hundreds of millions of dollars. Those motherfuckers couldn't even put my name on the fucking patent. Like, nope. This is everything that's wrong with fucking
That's the
America.
way it worked.
Yeah, dude.
Well,
I hate that.
from the other side of it, you could say that you never would have been able to figure that out if they didn't give you the millions of dollars worth of equipment.
Oh, they didn't give me $1 million. The way I figured it out was fairly rudimentary, but they had I spent a couple other dollars for sure, but not that much. But I get the whole intellectual property, or hey, we pay you to do this. This is why, for me, dude, I was like, I want nothing to do with that. I'm fine.
You're an employee at a company.
Right.
This is why I also didn't wasn't too keen on going
Yeah.
into management or anything like
Yeah.
that. It's that you realize that and
Yeah.
you chose not
Yeah.
to be involved in that system. And so
Yeah.
I can't really blame companies and say that that's a
Sure.
bad thing.
I get it. But put my
I
fucking
mean,
name on that.
it's the same at academia, dude. They made us like sign things for the university. If we ever published papers that generated any income for the university, it was their property.
Yep.
This is just the way the world works.
I get it.
It's not only like companies. I mean,
Hey,
companies
I don't gotta like
money.
it.
Companies are designed
Sure.
to make money.
Yeah.
So if we just destroyed that whole system, America wouldn't be America.
That's true. That's true. But put my name on the fucking patent. That's all I'm saying.
Well, if you
be
could
a
go,
total
you could
asshole
sue
about it.
them. You could go sue them if you want.
I mean, what? They already got, they had 7 attorneys just for the bye-bye motherfucker meeting. So, yeah. And they're Thiokol, that's the company that made the O-rings that failed on the Challenger. It's a major French company. It's a massive aerospace company. I can't ever sue those guys. That's never happening. Plus, they have all the, like, I have no evidence. That was all kept with them. That's all, in-house. I would have no way of proving anything at this point.
Yeah, that's the reason why when I left my last job at 5 p.m., they promptly disconnect. I couldn't get back in the building.
Yep.
Because there's... Yeah, I can see it from both sides. I mean,
Sure.
like if somebody comes to work at your mushroom farm and like learns all your kind of secrets on how to do like karyotyping or ISSRs or whatever. What if you figure out the primers to like do paternal testing for cubes, and somebody just happens to look and take a picture of your primer sequences and walks out the door with them.
Yep.
You spent 5 years developing these primer sequences. All of a sudden, like somebody's just got them.
Yep.
Or what if you create a new, cube that's like, I don't know, 4 or 5%, 4 or 5% psilocybin, and somebody just buys a liquid culture off you and then expands it and makes $1 million off it.
Thank God that isn't happening in our community.
Oh, it will. This is the thing. It's good to have a sense of like paranoia. Like everybody is out to fuck you.
Yeah.
The whole world is transactional.
Yep.
This is what you, especially living over here, you realize that every friendship, every relationship, every binding legal agreement, every job, it's all transactional.
Yeah.
People want something from you, and they generally have something you want from them. That's when the faster people realize that, the better.
Agreed. Yeah, dude, that happens in nursing. A lot of the hospitals will pretty much cover you. They'll say, "Oh, you don't need to buy nursing insurance. We protect our nurses." Well, sure. Until they got to throw you under the bus, and then they don't protect you. So yes, buy your own fucking insurance, because they'll protect you so long as their attorneys are advising them, like, yes, we can protect this person. And they want to, they don't want to look bad. But yeah, I've seen a couple people go down pretty hard, and they were just like, wait, I thought you were going to protect me. And nope, they got left high and dry. Yeah.
So they need a scapegoat or something. Yep. Yeah. The people my coworkers that I thought were my buddies for 18 years, threw me under the bus.
To protect themselves?
Oh, yeah. Yeah. They blame you for things. It was like the other guy's student started a fire one day in the lab, and because I happened to put it out, all of a sudden I was like responsible for it. And I was like, wow, that was like your students. I was just like walking by, and it was like he didn't want to get blamed for it because he wasn't attending, he wasn't taking care, he wasn't watching his students. And so all of a sudden I became the supervisor because I just happened to go in and put the fire out. It's like, wow, I should have let it keep burning.
Yep.
Yeah, there's the hundreds. I'm sure everyone's got hundreds of examples of getting fucked in life.
Yes.
Again, just talk.
Sometimes it's figurative and sometimes it's literal too.
Yeah.
Yes.
Yeah, at the same
100%.
time.
Yes. Sadly, yes. Yeah, man, that's really true in this space right now. I had somebody wanted to do some business with me, and it was looking real promising. And I think I told you about this, but we don't have to go into details about it. And we got to the point where I said, all right, cool. Let's start talking the partnership agreement. Let's talk about what bank we want to set up our business at. Let's like really do it. And the minute I did that, minute I had that conversation, it was just immediate ghosting. Didn't hear from the guy for months and months. I finally messaged him back going, "Hey dude, like you just kind of dropped off the face of the earth and you came in real hot and heavy on this. What happened?" And he was like, "Oh, something came up and all that stuff." But yeah, people, it's hard not to look at that situation and go, it's interesting how you were in my DMs all day, every day until I said 50-50 and let's put it in writing and get it notarized. Then all of a sudden the person disappears. So yeah, guys out there, if you're trying to do business with somebody, I don't care who they are. They can be your fucking dad. Get that guy to sign a partnership agreement.
Yes.
I'm telling you right now.
Yeah.
You cannot.
Some of my.
Yeah.
Those guys, when you find out the will, like you're not actually in the will,
Yep,
things
yeah.
like that. What he just talking about that the other night. Yeah, it's like, wow, you didn't know, like your mom forgot to tell you we're not in the will anymore.
Yep. Or worse than that. Worse than that, dude. My dad was friends with his two uncles. These guys never got married. They had inherited some family land, and they were the one guy was a little odd. My one uncle was a little weird dude. I'm not. Everybody's like, are they gay? Why are they living together? It's so weird. The classic stuff. But no, they were just like these classic curmudgeonly, tinkery, old coots that they worked a factory job. They had a family farm, and they just never had time to even look at a woman. They're just fucking workaholics, just work, work, work. And when they died, the weirder one died first, and there's no will, and everybody finds out in the family the guy had, I fucking shit you not, $1.6 million in his checking account. Both of them lived off the farm money, and their money from the factory they worked at just went in the bank. They just never did anything with it. They never invested it, never nothing, just went in a fucking checking account. And so most people ignored those two, but my dad was real good friends with them. And everybody's like, oh, said to my dad, you should get some of that money. They would want you to have that money. Well, my dad ain't going to go fight for that money. He's like a nice guy. And so who gets the money? The biggest piece of shit in our family. And if you're listening, you already know who the fuck you are. You are a piece of shit. They got all the money because they were willing to spend a few bucks on an attorney, go in and just clean house. So then when the older one dies, the one who lived longer was older, so when he died finally a couple years later, they were ready to go. There was, even then there was no will for this guy. Just the craziest shit I ever saw in my life. And again, they got all the money. Nobody else was going to fight him for it. I'm like, this is what happened. Just the wrong people got all the money. That is how life goes sometimes. The good people, the right people, they get dicked over by loser asshole pieces of shit. It does happen. So man, I saw that happen and I said, that shit is never going to happen to me. I am not ever going to get taken advantage of that way.
Happens all the time, dude. I got my own family too. I've chose to move on though. I don't want to dwell on that kind of stuff. That's the kind of stuff that'll eat people up.
Oh yeah.
It's like when I moved to Thailand, I don't even want to go into the details, but every family has those things. And I was just talking to a guy last night who's got basically his whole family moving over here. And he's going to be in this situation where, lawyers are going to get involved. And I don't know. You got to I don't know. People that dwell on that stuff, it eats them up.
Oh yeah, for sure.
It destroys your life. If you let stuff like that get into your soul, it'll fucking destroy you. I know 60-year-old guys who basically their whole life is fucked up because of something what like you just described, but they let it destroy their own life.
Yeah.
So you can dwell on stuff like that about who blamed you. And we were talking about how people really, really like to blame other people.
It's all that
It's way
matters these
easier.
days. At work, I see it at work all the time. Yep.
It'll destroy you,
Something goes
though.
wrong. All they want to do is know who the fuck did it. They'll even sit there, and I'm sitting there going, we gonna worry about the patient, or y'all gonna just go over there and worry about who fucked it up? Can we worry about who fucked it up later? Can we deal with this patient right now? They don't want to do that. It's all they care about. Whose fault is it? Yeah. That's how people work. They're ready to blame somebody. They want to play that game. I mean, we're living that. I know you're not in America right now, but, you know, that's what's going on in America right now. We're just, you know, both sides just taking turns blaming each other. You know, when it's like, nope, it's our country. It's all our country. We're all doing this. We're all part of this. Let's try to dig ourselves out of this hole. But no, we're too busy just taking turns blaming one another. Which is why when you think taxonomy is complicated, try to run a country.
Yeah, this is exactly why I
Even
don't
more
live in America.
complicated.
This is exactly why I don't live in America.
Yes.
I choose to exclude all that stuff from my life, and I lead a much happier life because of it.
Yeah, man.
So as an individual, you can make the choice to let that stuff affect your life or not. And I choose not to, so
I'm with you.
up to people. I get it secondhand from people, and I see what it does to other people and their lives and their mental health. And I'm like, why are you letting other people control your life?
Yep. Well, and half the time you're just talking about shit that you're not doing anything about. You can't do anything about. So why you spend so much time talking about it? Yeah. Just not really solving any problems. Yep. I hear you.
It's just making you die faster, get higher blood pressure, eat more, exercise less.
Increase cortisol levels. Yep.
Yeah,
100%. Stress kills.
Yeah,
but that's what people like.
Yeah.
Yep. All right. Well, so hey guys, we'll do the segue before we get into the second paper. Just so everybody knows, I got it's not done. It's up. I decided we're gonna start doing some giveaways on the show every week. So go to mycolgeeky.com. I've been sitting on this frickin' domain for like a year and a half, paying Squarespace for it, and never published it because I never got it done. And I finally just said, all right, we're just going to get something up. Go to mycolgeeky.com, sign up for my newsletter, and you will automatically be entered in giveaways every single week. So you sign up once, you've got a chance to win every week in perpetuity as long as we're doing this, which is if my plans go the way I want them to, we're going to be doing this. We're going to ride out into the sunset doing this podcast as long as it makes sense. So you go check that out, get signed up, and we'll be giving away genetics. We'll be giving away merch. We'll be giving away all sorts of fun stuff. It will evolve and hopefully get more and more interesting as time goes on. You know, maybe we'll get Ed. Ed likes to send me some stuff. Maybe we'll get Ed in the mix here. Ed, I didn't talk to Ed about this, so yeah, we'll have to talk privately about all that. But yeah, so go do that, and we're gonna segue now into you know, if the first paper wasn't nerdy enough, we're gonna continue the trend. We're gonna stay nerdy. We're gonna go all the way into I think a realm of things that really apply to a lot of, I think there's a lot of people talking some ideas right now, and maybe they read a single paper, or they're seeing some things in their grows, and they're trying to figure out what could be causing it or whatever, and they just read a little tiny bit of something and they think they get it all figured out. And so this paper is a step in the direction of getting people a little bit more familiar with some of these ideas that are getting thrown around right now. And hopefully over time with Ed, we'll have a much greater understanding and be able to discern when somebody is talking out their ass and when somebody is, seems to be coming from a solid place. Before that, I made one of these things. I better frickin' use it. Let me find it here. All right. So guys, I always got people in the live chat reminding people to, hit the like and all that kind of stuff. So I made my little, even for the lives, we got this. There it is. This sounds nice. People like sounds. That's what they say on the YouTube videos. They like sounds like that. So there are your sounds, guys.
Oh
Go.
yeah, I noticed you had a few sounds, a little laugh track thing or something in there.
I added the dude, yeah. I'm trying to so the new intros, I'm trying to shorten them up. I was talking to somebody one time and I'm like the average first view time is like 30 minutes. So even though we do these, sometimes we're doing a 5-hour podcast. On average, most people when they start watching watch for on average 30 minutes. They come back and finish the episode. I get people who are like, "Oh man, that one you did with that was a long one. That took me a few days to get through." Blah 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 blah. So what my goal has been is just to get into the meat and potatoes of the interview quicker. So I'm never gonna be more than 5 minutes to interview. I've been getting it down under 3 minutes these days, which is the goal. Just try to let you guys know up front what we're doing. Just get into it. Just polish it up a little bit. So yeah, trying to crack a few jokes here and there, make it fun. Keep people watching is basically the idea. So if we get into the meat and potatoes of it and we start talking about wood rot fungus sooner, then people will go, "Oh my God, we're fucking knee deep in wood rot fungus right now. I can't leave. I got to listen to all this." I mean, I know he's going to tell a story about Ron beating one of his thesis, beating one of his students with his thesis or something like that. So yeah, that's the plan. Just trying to clean it up, trying to evolve. I was looking from 2023 to 2024. I felt like maybe the topics broadened a little bit. I was getting out in the woods more. We're doing more, field mycology, having different guests on, but I felt like the format of the show was not evolving too much. So this year, just trying to, always trying to switch it up, try new things, keep it interesting. So yeah, if you guys hate it, let me know. If you guys are liking the polish of the front end of the show, let me know. I appreciate hearing from you guys. And yeah, doing giveaways. We do them in the server off and on. And somebody was like, "You should do giveaways on the show." And I was like, "Man, I never even thought about that." Yeah, that's a good idea. So we're going to get going on that. We'll be giving away some genetics next week. So sign up. And the great thing about setting it up this way is if you don't watch this episode for 3 weeks and you miss out on next week's giveaway, it's okay. Get signed up. You'll be signed up for the future week's giveaway and all the giveaways going forward. So and get in now, because while the pool of competition is small in the beginning, your odds of winning are very good. In a year or two, when there's 10,000 people, to choose from, your odds go down. So sign up now, win some cool stuff, have a good time. All right, so here we go. So we're going to jump into this horizontal gene cluster transfer, increased hallucinogenic mushroom diversity paper. And it's interesting. You sent me this paper too, because I got to meet this guy, Patrick Brandon Metheny, at NAMA a couple years ago. And then Scott Astouni and some of those guys talk about Jason Slott quite a bit. So it was cool just to be nerding out enough to get a paper and be like, "Oh, I met that guy. And oh, I've heard about this guy. They talk about this guy." So yeah, that was just like a little private cool moment I had. But yeah, talk me through this paper, what you were, why you were looking at it, and what your thoughts are after reading it.
Yeah, this is an interesting point. I don't know if maybe I didn't send this to you. I wasn't familiar with this paper until you mentioned it earlier.
Do you have it? Do you need
the
link?
I mean, I know the base. I do. I'm looking at it right now, but I wasn't aware of this paper. I'd heard of another. I'd heard of horizontal gene transfer, and the evolution of psilocybin in nature. That one, what was it called? The ecology and evolution of psilocybin in nature. And I heard people talking about this paper, but I wasn't aware of the actual paper. I just assumed they were talking about that paper. So
Man,
Yeah, this idea.
my bad. I
Yeah, I was
thought
a
you
little
sent me
freaked
this
out
paper.
when I saw the announcement on your Discord. I was like, oh shit, like I didn't even know what you were talking about. I'm like, oh,
Oh
I'm supposed to
crap.
talk about that. But yeah, I am familiar with this idea that basically horizontal gene transfer. I mean, to be honest, I probably know more than like a lot of people about it. It's the idea that at the same sort of taxonomic level, as well as that at the same time, like, so you have two individuals that, like, say, November 2nd, like 1999, that they've decided to transfer horizontally genes, and they can do that through a variety of mechanisms with plasmids. So the simplest, or the one probably most well understood organisms, are bacteria. Like I was talking about, this conjugation and transformation and the sharing of plasmids in bacteria. These things have been well known for, oh God, probably 50 or 60 years. This idea that bacteria, because they don't undergo sexual recombination that often, or at all really, they need some way to transfer, say, antibiotic resistance genes. So, like working in the hospital, like let's say you get somebody who comes in with MRSA, methicillin resistant Staphylococcus aureus. So MRSA, and then next thing you know, you've got this outbreak because everybody has these normal, like, let's say, Staph aureus, Staph A, it's everywhere. So now you have somebody who comes in, a patient who's infected with this MRSA. It's this, like, resistant form of the normal Staphylococcus aureus, and then all of a sudden you've got this outbreak, and they were trying to figure out how this happens. There's other examples in, like, yeast, for instance. You have, like, yeast, like in wine making. You'll have like a batch of wine that's going great, and then all of a sudden you get these things they call killer yeast. And you get one of these yeasts in there. It's like the spread of sometime any other contagion. And so the idea is that it could be antibiotic resistance gene. It could be some, like, factor that decreases some metabolic activity, causes death. It almost like it introduces like a cancer. And so this idea that it spreads horizontally is the basic idea. Now, how this happens in, like, mushrooms and cultures and things out in the wild, that's the part I'm not familiar with. It's and the other thing I saw, like, sometimes is, so the idea is that it's horizontal, not vertical. So genetics are normally, you pass genetics on through, mother, father, son, daughter, that, like, vertically, like, so over generations, so over different time periods. So that would be the vertical
And through a
kind of,
reproductive cycle.
yeah,
Right, yeah.
yeah.
So
So that's the
horizontal means
normal
no
way.
reproductive cycle, but
Right.
somehow they're swapping spit. And in the spit,
Exactly.
there's some interesting genes. Yeah. All right.
Yeah.
That
So
that makes
swapping
sense.
spit, but not sperm.
Right.
Yeah, I don't know
Yes.
if that.
Oh, I
Yeah,
don't even need to put that
that's
one in
not
my
might
head.
not be a
Yes.
good.
Okay.
So
Yes.
so yeah, so it's like, maybe that's not a good, because that means you're doing other things besides just
Right.
swapping
Right.
spit.
Yes.
So how about you
I
rubbing
made
up
it.
again? How
I
about
made
you're
it sexual.
hugging? You're
There you
hugging
go.
each
There you go.
other, and then you somehow are going to transfer genes to that other person.
Right.
But if you're actually reproducing with that person and making offspring,
Right.
that's going to be vertical. So
That makes
horizontal,
sense.
literally, it's just like, if you look at it more from a temporal standpoint, at the same time, you're just simultaneously
Right.
exchanging genetic material. So that's
Now,
easy with bacteria, because you can put them on a plate and you can be like, this is a resistant bacteria and this is a non-resistant. And when I put them together, all of them become resistant. So this is the problem. So this could be with viruses. This is why viruses are so fascinating, is because you've got everything in the human. You got cannabis. So, like, when people are doing clones in cannabis, you take your scissors and you chop this clone, and then you don't sterilize your scissors and you go to the next one. Somehow these viroids are getting transferred, just like humans transfer
right.
viruses. People that get the cold, or the flu or whatever, like, you don't want to be near them. And
Right.
if you see somebody wiping their nose and snot and everything, you should probably stay away from them. So that would be like horizontal gene transfer, not in the same organism, but the same basic idea.
Right.
Whereas vertical, yeah, like you said, there's
So,
reproduction generally, and it's gonna be a more.
so now let me ask you this. To be clear, humans can't do this. I can't like have a really bad day and accidentally give you red hair if I'm a redhead, right? Like we're not specifically, is there a way mammals, for example, can do this? Or is that
No, well,
all
that's
epigenetics, right? Like maybe you could say there's some epigenetic things that could happen that could adjust your DNA, but you can't actually transfer DNA human to human in a way that changes them. Is that correct?
I don't know. I don't
Okay.
study mammalian
Okay. I
genetics.
just
I'm not
I
a
wondered if
profession.
that,
I don't
yeah.
know.
Okay.
So there's viruses that can be transmitted, and then I think
Uh
I have to admit I'm not an expert on mammalian horizontal gene transfer.
okay.
I barely understand what the terminology means in fungi. I'm sure there's gonna be things that'll probably like be discovered, but I mean we obviously transmit viral particles amongst each other. I mean there's like retroviruses and things that can go on, and
Right.
like I said, maybe that's epigenetics. Some of these terms I'm getting a little bit confused on where they apply these terms. Like epigenetics is just sort of this generic term that anything that's not part of the typical genome. So you then have your virus, bacteria, plasmid, plastids, bacterial mediated stuff. All of this stuff sort of merges together. And when it comes back to mushrooms, I'm a little bit confused, even when I've read these. So why I bring up the time thing is because they'll often say like, well, these psilocybin gene clusters were shared at some time in the evolutionary past, 10 million years ago, 50 million years ago. Like you can't really, it's almost like you're looking at a crime scene after the crime has already been committed and you're trying to reconstruct what happened, but there could be other explanations, you know?
Right.
Like if you see blood splatters on the wall, I mean, that could be that there was a murder there, or it could just be, I don't know, maybe I like smacked my head
Right.
on the wall, or there could be other explanations. But the only thing you can do is look at the information or the evidence after these things have happened. And I don't know what they say,
Right.
to infer what had happened in the past. And so these things will likely change. This is the way all of molecular systematics works. We're looking at 2025 DNA sequences and we're going back in time and trying to make us, we make a lot of assumptions and we're trying
Right.
to say what happened. That may not be the case. It's like you
Oh
just see
yeah.
this snapshot of what is occurring right now. Whether, cubensis picked up these psilocybin genes from a paniolus or vice versa, like you can't go back and there's no like a caveman back there with a petri dish and being like, well, let's see if I mix these two together. It
Right.
just doesn't
Right.
work that way.
But now, so because I read that one paper you're talking about where did this all come from and all the theories, and was sitting there going, well, tryptophan is everywhere, right? That's in a whole bunch of stuff. So then to go from tryptophan to what's the next one? Tryptamine? I forget. But in the process, right? You got to
For
take
psilocybin.
yeah, to
Yeah.
ultimately get to psilocybin, you got to go from tryptophan, tryptamine to another intermediary, and then you finally get to psilocybin. Um, but like, it's part of the paper. I'm sitting there thinking it could just be totally random, couldn't it? Like it could have just like does it have to have even had a very specific reason for it to stick around? What are your thoughts on that? Like, did it have to actually serve a purpose, or could fungi just do a weird thing and it just did it for whatever reason? Because these theories about, oh, it's to encourage or it's to discourage things from eating it, or it's to encourage things to eat it. Could it have nothing to do with that? And again, I
Yeah, well, of
understand we're all
course.
speculating. I mean, I know we don't know and we can never really know, but
We know a lot of people want reasons for stuff. It makes life simpler to
right,
understand when you have a reason for something, but
right,
oftentimes there's no reason for something. So you can go back to the idea of natural selection and that, like, if this is not a advantageous characteristic, why would the organism keep putting energy into making psilocybin if it doesn't serve any selective advantage? Like if it's not a deterrent for herbivory, or it doesn't, probably have some even minor metabolic, maybe reproductive enhancement or something like that. Or, I don't know what possibly,
Right.
but that's what that paper was all about. And so people have been speculating about that for probably ever since they knew about magic mushrooms. I mean, Gordon Wasson going down to Mexico, coming back and being like, look, they're these mushrooms that like fuck us up. Like, of course, you got to have a reason for it. I mean, the first publication in Life magazine, I haven't read that in a long, long time, but I'm sure he speculated that, maybe there's a religious significance of it and things like that. I don't know. People need reasons. I don't want to, like, people have to have a reason, like for good and evil, right and wrong. And we have books that tell us what to do. And that's the way people like to live their lives. People get very, very uncomfortable when they don't know the reason for something,
Right.
or they
Yep.
don't know what happens next. It makes them feel very, very uncomfortable. So whether there is a reason or not, they will find a reason or propose a reason.
Right.
The reason for I'm not that smart, and I don't think anybody is to even be that I don't know, that arrogant that we would think that there's some understanding of why Mother Nature made psilocybin. There's a lot of different ways you can look at it, but those are usually very biased by one's personal experiences, maybe.
Right.
It's like when I'm tripping, I generally just think like, wow, this is really fucking cool. But somebody else might be like, oh wow, is this like I'm talking to some higher power? And when you first start your first few trips, you're like, oh, is this like the way I talk to God? And then after about 100, you're just like, nah, this is pretty cool. Like, I'm looking at that thing on the wall and it's moving. Like, this is pretty fucking cool.
Right.
Wow, my brain's pretty simple. Like, I ate these things and that little like serotonin pathway on my brain just got all fucked up.
Yeah.
And then you start to think like, well,
Yep.
maybe this isn't that really significant. It's like, I don't know, like the first time you drink a few beers, you're like, wow, I feel different. Like nobody's thinking like, well, this is like well, they're probably thinking this is the way I find a wife, or this is the way I make babies.
Right.
But but I don't know. A lot of people, they like to put a lot of meaning in stuff. I'm not really one of those people. I kind of take stuff at face value, but as far as like figuring out what psilocybin does in nature, I mean, papers like that are very inconclusive.
Right.
Those people are probably way smarter than me, and they didn't really figure it out. So I don't really think about it that much.
Yeah,
I've
man.
simplified my life a lot. I just don't think about things sometimes. I choose not to really think about a lot of stuff anymore. It makes life way simpler.
Yes, especially if you can't ever know the answer.
Yeah,
Yes.
you gotta frustrate the hell out of
Yes.
yourself,
It's
and
really
then it's
true.
like I like to learn about new stuff, but I there's a point at which you're not gonna figure it out, so you
Yeah.
might as well like kind of ease back on the I don't know,
Yeah.
furiousness of how you which you approach some topics.
Yeah.
But I get a lot of people sending me these messages like, and they're asking me that same question. I just had a guy yesterday who was really fucking hammering that fucking thing down. Like, you have to tell me what the purpose of psilocybin is. And I'm like, dude, you need to fucking chill out, because I sent you the link to the paper. Send it through. Like, I don't have the answers, man. Go talk to your fucking preacher or whatever. Like,
Right.
I don't got the answers for you, dude.
Now, isn't there's also chemicals that have no effect on us, but then affect other animals, right? Like catnip, or doesn't eucalyptus get koala bears high? Like there's a bunch of these things where, so, I guess if the koala bears could talk, I'm sure they would sit around and as they're like munching their eucalyptus, they'd be like,
The
"Hey,
eucalyptus gods.
what? Who did this? This is great. This must be for me, right?" Yes. I think you pretty much nailed it. I think it's because we want to know, because it is so cool. It is such a novel compound. It does cool things for us. And of course, we want to just know, why is it there? Why is it in the mushrooms? And we definitely put a lot into that whole thing. And who knows, one day, Ed, it might be revealed to us that we are all just a designed hologram from the alien sentient beings. And then we'll be like, okay, look at they programmed Ed to think what he thought. They programmed me to think what I thought, and it didn't matter anyway because you know, they designed it all, or nothing, or it just happened. And like you're saying, we really don't have to know exactly why. What implication does it have to know? If you're not getting a PhD, or you're not trying to, you know, keep your job at institution X, you might really not ever need to answer that question. And the implications for us growing mushrooms that have psilocybin, that have the Psy-K and Psy-M and all these little genes that help synthesize this great little compound we love. That's how it works. You know, I'm not sure there's a way to hack those genes specifically to make them make more or less or whatever, but
there is.
Make it, like, would you just make there be more of that gene expressed in more places on the genome?
There's a variety of ways to do it. I don't think that we'll do. That's getting way out in the weeds. We're gonna lose people.
Sure.
So yes,
and we don't need to do it anyway because we got. I mean,
we
got plenty of mushrooms. We're good.
There are ways to increase the expression of certain compounds or enzymes. It's very well established. Okay. Yes. Yeah. So it's just like if you make insulin, they've already done it in yeast. Yeah, that's well established. But I think that's beyond the realms of like the home cultivator. If you just want to grow yeast in a bioreactor to make psilocin, you can do that. They already do that. I don't have any particular interest in that. If you want to overexpress the psilocybin genes in pubensis that you grow at home, you could do that, yeah. Again, there's no commercial application because you can do it more cheaply in yeast. Right. But that's easily, if you want to do that, I could download 3 papers right now that tell you how to do that exactly.
All right, guys, there you go. There's your FAFO for the weekend. Go.
Yeah, I mean, it'll cost you. You'll need about probably $40,000 to $50,000 worth of lab equipment and probably a year to do it. Right. So a lot of people are just going to want, like, the same thing. If I stumble across a 5% cube, the easier way it is just get the yeast.
Sure.
Like people used to go to the Guinness brewery and get the Guinness yeast instead of trying to develop the yeast yourself for hundreds of years of breeding. You just go get the yeast and steal it from somebody.
Yep.
So that's a lot easier way to do it. So just wait till one of those undergraduate who works in that lab that uses that psilocybin yeast and managed to just sneak some out of his pocket. He'll be selling it on Etsy for like $5 a sample. So don't bother doing it yourself.
Oh, that's fun.
So that's where we can take advantage of the companies. You know, you get fucked by the companies, but you fuck them back. Because as soon as somebody walks out with that like yeast or E. coli that produces, it's.
Right.
Yeah, it goes
or all we got to do is just get one guy to go work for these people. Yep, and there you go.
Oh yeah. Within 5 years, that yeast will be widely distributed.
Yeah, and then you're just gonna use it for your bread and for your beer and all that. And the next thing you know, you're tripping balls on your beer and you're tripping balls eating your ham sandwich at lunch. And we can just there you go. Who knows? Sky's the limit.
So there's other issues with that, but I don't think we should go down that.
I'm teasing.
It's not going to be like that, for a lot of practical reasons, but back to the thing about so this pathways are ubiquitous. So I think as far as like the humans, like trying to figure out what psilocybin is in mushroom for is almost kind of pointless because it's there for one, and two, it's like these pathways are fair. Like you said, the tryptophan, it's an amino acid. It's all over serotonin. I mean, it's a ubiquitous. It's a plant hormone.
It's like for
it's
protein,
the basis.
Right?
Yeah. It's an amino acid that's ubiquitous in nature. It's not, I don't think anything. It could just be like the simple fact that maybe there was some little mutation and this thing gets created. I mean, we still have an appendix and all these other things that we don't really know exactly what they're for.
Bro, what if our appendix was where we made the psilocybin, bro? Oh my God. No, yes. Yeah. A lot of things we don't know. Why do we get hiccups? They haven't even figured that shit out yet either. Yeah. Who knows?
Well, that's an imbalance in carbon dioxide in your blood, isn't it?
Oh, my
I,
understanding was they didn't really conclusively know what that was,
oh,
but
I
I
don't
know
know. You
that
would know.
is one of the theories, and one of the others is that it's an innovation from ancestors way back that's really just redundant or not used, and yeah, who knows?
Oh,
I don't know. Yeah, maybe it's that.
oh, I
All
don't
these
know.
little weird things about ourselves that we don't know why we do it or have it and all that stuff.
Well, that's why we need more people. This is, it's one of those things we've talked about a lot of this stuff before, and I don't like at the end of the day, nobody really like a lot of people don't really care that much. I mean, we've kind of had this thing when we've gotten to some of these deeper topics before, and a lot of times there's a certain very topical level of interest for most topics these days. And once you get past that sort of just like how can I get more high? Like
Right.
people lose interest really quick. Like,
That
oh,
is
you're not.
the
I don't know how to like. I want oyster mushrooms that I can get high. Like, and once you don't know how to, you don't tell
right.
them how to do that, they quickly lose interest.
Yes. That is the primary interest for sure. Or I feel like really there's still an opportunity for a podcast or show on YouTube where all it is just people presenting a picture of their tub and just asking, is it contaminated?
Hmm.
Feel like you could do that for hours and hours and hours. Never ends. Every day, I usually have somebody asking me that question. Yep.
Yeah, it's like
Yeah.
yeah, I get a lot of those too.
Yes, it is pretty common. Yeah.
Like,
I will say this. I think there is we're in this spot with the community right now. There was such a flood, and I think still is a big flood of people coming in, thinking there's some money to be made here. I'm gonna tell you right now, go get a job at Target. You'll
mm,
make more money there, and it'll be easier money. I every day hear about somebody else stabbing somebody else in the back. People
yes.
getting really gross and nasty, and at the end of the day, I think what a lot of people forget is there's plenty of people paying attention to how people are behaving in the space. And they might not be saying something to your face, but if you're doing dumb shit and behaving in a way that's not ethical, people see that and it's not doing you any favors. So shape up, or it's gonna have an impact on you. If you truly love to grow mushrooms and you really truly need to be in the space and you can't imagine a life of folding clothes in soft lines at Target or working at a factory or doing, you know, like scraping shingles off of a roof, whatever, job you're trying to avoid doing by doing mushrooms instead, man, it better be the absolutely only thing you want to do because I know people have been doing this for 20 years, 10 years, 15 years, and they're quiet. You don't know who they are. They're talking to me, telling me what they're up to. And those people that have just been quietly earning a living in the space for a long time are telling me it's not what it used to. I don't know how much longer I'm going to be doing it. And if the pros of decades are saying that, and you're brand new and you just learned how to grow your first tub of mushrooms, I don't care how enthusiastic you are. Just the reality is, it's probably not the wisest play. Grow mushrooms for fun. Grow mushrooms
Mm.
because you want the medicine for yourself. You want to give it away to some friends and family, but you don't have to instantly commercialize, a hobby or an interest.
Yeah,
Just
I
don't.
hate to say
Yeah.
I hate to tell people that too, or like feel like you're discouraging people, but it's just the practical the reality of it.
Yeah.
It's like you gotta realize that these things are all gonna be taken over by large companies at some point. And
Yeah. Oh
either
yeah.
gonna
All the
be
legalization. The minute it's legal,
Yeah.
I mean, at that point, all y'all are going to get to do is grow mushrooms for yourself. And
Hmm.
even that might be in question at a certain point. But yeah, hoping for decriminalization is probably my greater interest. But enjoy where we're at right now, or at least where we've been. It is getting a little bit uglier. Man, just grow mushrooms for yourself. Make a difference locally in your community. If everybody just focused on that, then we would really we would be making a positive impact on the rest of the world's opinion of us and what we do and why we like this stuff, and how the stigma that's there really doesn't exactly make too much sense.
Yeah,
Try,
exactly.
yeah. Try
We
to
need
try.
to improve our image, I think.
Yeah.
Because
I'm
there's a
with you
lot
on
of
that.
people, professionals especially, that don't want to be associated with. I mean, let's be honest. Like, they don't want to be associated with a bunch of drug dealers.
Yeah.
Like, if you're a clinical psychologist or whatever, a pharmacist or something like that,
Yeah.
you gotta keep your distance from these people that are, I don't know, maybe the
Yep.
unsavory elements of society.
Yep.
And
100%.
so it's gonna inevitably go to the big companies who have what are SOPs, and they're gonna have safety procedures and things. Like, so I hate to say it, maybe this is gonna make some people butthurt or whatever, but I made a post the other day about, and somebody else was talking about the contaminated tubs. It's like if you were a business person and you wanted to sell a product and you're looking at some of these like grows that people have, you're like, there's no way, like, I wouldn't eat that stuff. It's like if you go to the market and you see rotten apples, you're not gonna buy them.
Oh,
Like, and
yeah.
I
Oh, dude, I have people who show me a picture of something and I go, kind of looks like there's some, it's fighting some Pseudomonas. Here's evidence of X, Y, and Z. There are some telltale signs of Pseudomonas infection. And their very next question is, but can I eat it?
yes,
Sure. Sure you can. Go for it. I
we have
wouldn't,
such
but
low standards.
but okay, fine, go for it. Or they got a giant green patch of trichoderma next to two fruit, and they want to know if they can eat those two fruit. Sure, man. I don't care.
Oh,
Go for it.
I
Whatever you want to
understand.
do. Whatever
I
you want
used
to
to
do.
be there too,
Yeah.
long time
But
ago.
that rite of passage, you can tell them, but until they get there, they don't realize that you go from just the scarcity mentality when it comes to fruit to just overwhelming abundance, right? Until you get
Mm.
it figured out, you're just like, God, if I could just grow a single mushroom. And then one day you're like, I don't know what to do with all these mushrooms. I
Yeah.
can't get them away quick enough. Yeah,
Mm.
it's really true. It is really true.
Yeah, that's why I think we need to just, yeah, spread knowledge amongst I don't know,
Yeah.
try to make other individuals' lives better.
Yeah.
Like, don't try to think about it like you're gonna get rich, but I understand also it's generally young guys that maybe don't have a super duper career going or whatever, and they're it's kind of like they
Right.
just think it's the Scarface kind of movie thing. It's like a cliche, you're
Yeah.
coming up and you want to, like, all of a sudden you get,
You got something to
delusions of
prove. Yes.
grandeur. Yeah, like, I'm going to be a big shot. And uh, it's a lot of guys go down that path. I mean, maybe some girls, but it's usually guys.
Yep. Yep. Yep.
And yeah, luckily they won't, I mean, at least these days they don't end up in prison anymore so much. But
Right.
if you think you're going to get rich, you should
Man,
probably like look at the track
I
record of a lot of other
yes.
people before you.
I have said this, I think, on the show back in the day. I'll say it again. It's one of my favorite little stories. One guy told me a long time ago. This is a guy, one of these guys, he has got an extremely low profile. He's a bulk grower. He is not here for the limelight. He's got work to do. He doesn't have time, but we talk from time to time. And the best thing this guy ever told me, he goes, Geeky, if you follow, the trail of cocaine back to the source, you end up seeing a guy sitting on a giant pile of money with gold chains around his neck and a machine gun in his hand, and he's like a drug overlord of a Pablo Escobar type of situation. He goes, if you follow the chain of mushrooms back to the source, he goes, it's a guy in his mom's basement who can't even pay rent. And he said that, and I cracked up. And sadly, there's not, there's a couple people have figured out how to make money with grain and sub. You can definitely make money growing gourmet mushrooms if you're willing to do some work and find the right buyers. But cubes, especially these days, especially now that there's 1,000 products to make it easy to grow, there's all-in-one bags. There's people like Ed Grand and Myco Geeky telling everybody, all the tips and tricks and secrets, and have all these people on the show. If you can't grow mushrooms right now out of your house, I don't even know if you should be allowed to drive a car. It's really not that hard to do if you just pay attention and give it the old college try and learn from some mistakes. You can absolutely do it. But some people, they don't want to do the work. They go, oh, wait a minute, this is work? Or, oh, if it's not going to be work, it's going to cost me money? Then they don't want nothing to do with that. Those people don't do well. And then sometimes those people turn into culture vultures. They're like, well, I suck at growing mushrooms, but maybe I can just buy everybody's shit or steal it and then just start slinging it out of my back of my car. Yep.
Those people will be the customers for the big pharma and things like that. I mean, at some point, it's just going to be another pharmaceutical medication, probably, that you buy from or you get prescribed from your doctor. Might take 10 years. It might take 15 years. But
they're going to figure out how to turn it. Yeah, they're going to figure out how to turn it into a drug you got to take every day, though. Right? They don't want to sell a drug where you can trip balls once a year or once every couple, every few months. They don't want to sell 3 pills a year to you. They want to sell 365 pills a year to you. So I am hoping that aspect of it will maybe make it, as soon as it's legal, Pfizer's not going to start growing mushrooms. They're going to figure out how to do some synthetic derivative and come up with other applications. I feel like we're still going to get to grow our mushrooms and use them the way that we've been using them for, half a century, but who knows?
It's the old kind of Boy Scout prepper mentality, It's like if you know how to grow your own stuff, same thing, grow your own vegetables, grow your own animals.
Yep.
And if you can grow your own mushrooms, you can become independent of, the big pharmaceutical machine or whatever.
There you go.
And it's also the hobby, right? The hobby. I think people often overlook the people that just want to make money doing this. They overlook the therapeutic aspects of just growing mushrooms, like the hobby. I think that's like often overlooked. Most people, I think, when they get into it, they probably are. I mean, most people are probably thinking they're going to make a few bucks, or maybe they just want to get high or whatever. But then once you start actually growing and you see the therapeutic aspects of just, I mean, there's the other end of that too. If you want to be a farmer and make money, like if you really think you want to make money, you're gonna have to be a farmer. You might as well be a corn farmer. You might as well move out to Nebraska and grow wheat. Like being an industrial scale mushroom farmer is not really like what most people want to do. If you watch a lot of the gourmet people on YouTube or whatever, they get this idea that like, oh, I'm gonna make grow shiitakes in my backyard, and then they do that for a year or two, and they realize that, wow, you can import shiitake blocks for like $4 a piece, buy the container, and I can buy like dried shiitakes through an importer for like $2 a pound, and then it's like the whole profit margin just disappears.
Yeah.
So a guy was selling a mushroom growing container for the other day on Facebook for like $30,000 or something. It's just, like, how many mushrooms you got to grow to make back $30,000?
Oh my God. Well, cordyceps, you only got to grow like 4 pounds of them, but yes, you got to grow everything else. You got to grow
here they sell them. If those people that are selling cordyceps in America are fucking robbing people. sell cordyceps here like big packages for like $1.
Wow.
Cordyceps militaris. The only reason that it hasn't caught on in the U.S. is because the growers in the U.S. are keeping the market artificially high. So they're doing like every other
Right.
money
Sure.
person does,
Yeah.
is they're restricting the supply. So it's artificially inflating the prices.
Right.
Like
Well,
diamonds. Diamonds are actually quite ubiquitous in nature. They're common, but people like De Beers have made them special. Diamonds are
yes.
not. There's nothing, absolutely nothing special about
God damn
diamonds.
it, they're forever No, I'm kidding. I don't care. Yes.
Yeah, forever in debt because you spent 3 months of salary before you got married.
Oh,
That could have
these
went to a
days.
more tangible thing like a house payment or a
Right.
car, but
Yes.
instead
Oh
you
my
bought a
God.
piece of carbon to show how you bling bling how much you love someone.
Yep. Exactly. Somebody
Stupid.
just said, Wallsteb just said, he's talking about synthetic psilocybin. He said it's good, but definitely different. Have any of y'all had the synthetic? I will say this. I think another guy in the chat was part of one of the early studies, and he basically said the same thing, which was like, oh, you can definitely tell the difference between psilocybin you're ingesting from a mushroom versus just this purified psilocybin. So hey, maybe our mushrooms are have an edge up. Maybe they always will. That's definitely my hope. I,
Oh,
however, I'm excited that there might be some novel things that big pharma can figure out. Applications for traumatic brain injury or very targeted use situations. I
mm,
don't. I hate big pharma in the sense that I hate how driven by money they are. But I mean, there are drugs that have saved a lot of people's lives. There are, absolutely,
mm.
you can eat just so many turnip roots and fucking powdered leaves from the woods. There's a point where these days you end up in the hospital and you get big pharma drugs and they stabilize you and they solve problems and all that. So I would love to think that if it's ever legalized, cool shit would get discovered and figured out. But of course, will all get exploited in the process. But like Ed said on the top of the show, can't do anything about that, Myco Geeky. That's the way the world works. It's a profit driven world. So
It is.
yeah,
I mean, I know I talk about like, dude, I hang out with like old white dudes all day, every day, and I just want to tell them, dude, like, let it go. You're killing yourself. Like you're sitting
yeah,
in a bar drinking yourself to death because of
yep,
all this shit you're worried about. You are doing more harm to your own self, like worrying about this stuff. And it's like that's what big pharma wants. Like they want you to be stressed out. They want
right,
you want you to be like high blood pressure, eat more, diabetic, and like all this shit. But before we go move on, one obvious reason why synthetic psilocybin would be different because it's a single molecule.
right,
I mean, we all know about the entourage effect and the ensemble entourage effect, whatever
yeah,
that, the synergism piece. I mean, eating a raw mushroom with like 5 or 6 different alkaloids in it, it's going to be a different effect than pure, straight psilocin or
right,
psilocybin. I mean, that would be an obvious reason why they're
yep,
different. But
yep,
yeah, then it goes back to also the problem of, clinical trials. The way Western medicine works is you have to isolate a single compound,
right,
and that goes back to this idea of growing kind of consistency. Like if you grow something and I grow something, we can't call those the same.
right,
So if you're going to say that these medical benefits are attributable to like the thing you grow, it's got to be the thing you grow the same way every time. That's not
right,
going to apply to the thing I
yep,
grow. Maybe I have a different isolate. I have a different conditions. And so, yeah, and then it goes back to all types of problems with Western medicine and whatever. But
yeah.
that's why you should just grow your own mushroom.
You should just grow your own. I mean, I can tell you that every time I have to do CPR on somebody who comes in dead and we we play the game, we do the tricks, we can revive them quite a bit. But you're like, cool, you didn't have oxygen to your brain for 15 minutes. Guess what's going to happen? We're going to spend $1 million trying to, keep you alive. And then finally your family's going to be asked, to pull the plug at a certain point. And I'm just sitting there going, why are we doing that? Especially, sometimes we revive people and they get mad that we revived them from their high. Like they OD'd on heroin or fentanyl or carfentanil, and they literally fucking hate me for saving their life. All right, bro. I mean, I'd be ready to be like, here you go, here, have at it. You do what you want to do. Well, what am I doing here? Wasting my freaking time.
Or you like ruin their buzz.
Definitely disappointing to have them when they get that way, where they truly, you walk in and they got a look of hatred on their face for you, and you're like, I literally helped save this guy's life, and this guy is treating me like shit. He just wants to be high again. He's conspiring to get out of this hospital so he can immediately go do that again.
Yeah.
And man, half the time that happens, not even half the time, a good chunk of the time, it's the guy they usually get their heroin from, didn't have any and they had to get it from somewhere else, or somehow the usual situation didn't work out. Maybe they're in where I live, out of town for something, and they score from somebody else, but something different happens, and all of a sudden, or even their dealer has to get it from somewhere else, and it's the wrong consistency, the wrong concentration, the wrong whatever, or they think it's fentanyl and it's carfentanil, or they think it's heroin and it's laced with this, that, and the other thing. Whatever it is, it fucked them up and just about killed them. And I'm just sitting back every day this happens thinking to myself, yeah, grow your own drugs. Just grow your own. You know what you got if you're doing it yourself. Don't know how else to put it. Take care of yourself. Grow your own drugs. I mean, maybe you don't got to grow your own weed, but you probably should do that too.
Every time I watch one of those documentaries, I saw one the other day about Phoenix, Arizona, and they have these like fully functional like places where people can go live, but they got to stay clean and they can't stay clean. So these people rather sleep out under a bridge.
Yeah.
Well,
so there's a new initiative. There's a new initiative for that. It's Housing First Initiative. They used to have all these rules for these people. You can't drink. You can't do drugs. You got to be in by a certain time. You got to do all this stuff. And, these people couldn't even go apply for a job. They're like, everything about that system just didn't work. So they've had to revise that in many places. That no longer applies, but yeah, there are still places where, sure, we'll feed you and house you, but you have to follow these 19 rules that pretty much we know you're not going to follow. So yeah, go figure that they have funding, they have federal or state funding, or they've received donations, charitable donations to run their nonprofit that houses these homeless people, and they got empty beds all the time. Because their policy around who gets to sleep there is a little fucked up. Yep. Just put it on the list of shit that don't make sense.
I'll let you guys figure that out.
Yep. Not for me to figure out, that's for sure. Yeah. What's really too
they
bad...
have the same problem over here. It's the same, except for it's a tropical country, so people can just sleep outside.
Right.
I mean, just like 2 blocks from where I am right now, there's like 3 dudes living under a bridge that are... like, I don't know. They just live down there. Nobody really. I don't know.
Yeah.
They choose. I guess they choose to live there. I don't know. It's like easy. I guess. I don't know why.
I mean, I took care of a lot of homeless people in LA when I worked at the hospital, county hospital there. And, I mean, easily 100, 150 homeless people. And I don't think any of them, man, they would tell me some sad stories. I don't know if anybody like makes any like really well-adjusted person that has everything going for him says, this is fine. But what I'd rather do is never bathe, never get a haircut, never cut my toenails, never change my clothes, get raped, get beaten on a regular basis, and not have a job, have no self-esteem, and get addicted to drugs. I am not utterly convinced that that is anybody's life choice. I do think there's a point where people with mental illness, maybe they're like, I can't keep a job. My family has shunned me. Nothing's working out, so I guess I'm gonna live under this bridge now. That's my guess on all that. Who knows? Maybe there's somebody somewhere that truly made it a life choice, but I don't think too many do. I think it's a pretty bleak situation. Now, I will tell you this. I know that some people, for whatever reason, find themselves homeless, and it's such a fucking traumatic situation that if it doesn't get resolved quickly, it can trigger mental illness. It can trigger traumatic situations that then cause them to want to start doing drugs or drink or both. And it's just sort of an ugly, vicious cycle. All I know is it's so much easier and cheaper to keep people from becoming homeless than it is to solve the problem once they become, remotely used to that situation. It's unfortunate. But if you're going to be homeless, don't be homeless in New York City. It's fucking, awful in the winter. Yeah, go somewhere warm. Go to Thailand, homeless people. Just go hang out under Ed's Bridge. And it's warm and balmy. It does rain from time to time, but yeah. Yeah, it's too bad. These are situations that when we were living more primitively in tribes and extremely small clusters of people, the situation was easier to handle. The problem now is this issue congregates in big urban areas. It just becomes like a literal problem. You just don't see it in a small rural town, right? Like people hide their problems. They just let the guy sleep on the couch, whatever. Somehow it's not quite as ugly a situation in Farmerville, America than it is in New York City or LA or something like that. Not that I have an opinion about homelessness or anything. Yeah. I wonder, we should do a TV show. We just get a bunch of homeless people. We let them trip balls, and we just see like what that does to them. We could like do, it could be like a makeover show. You could get them, you get them the stylist. They get a haircut. They get a facial. They get the manicure, pedicure. They all that stuff, and then they get to go to a float tank, trip balls, integrate their experience afterwards, and see what that does for them. I wonder. I think that'd be a good TV show. I don't know what channel we'd put it on, but I think that would be interesting. What do you think, Ed?
I'm gonna let other people figure that out, dude. I actually purposely don't watch like those things that much because I all it does is depress me.
I know.
Yeah, it's one of those things again. I can only control what's in my little life sphere. Yeah. I can't control what other people do. So I choose not to think about it. I don't know.
And you know what?
It's selfish or whatever.
That is it's not, because you're not. You're not. If you thought about it all day, versus if you never thought about it at all, does that have any net impact at all? No.
No, exactly.
It only makes a difference to think about it if you go do something about it. My wife works in that sector. I hear about it all the time. That's the only reason I have an opinion about it. If not, I probably would be thinking a lot more like you. Just what do you want me to do about it? I can't save all the homeless people. Yeah. Yep. Anyway, all right, man. Well, this was nice. I'm sorry. I thought that one paper I thought I got it from you, but I guess not. I just assumed that if I got an interesting scientific paper, I got it from you. Must have got it somewhere else.
Well, to be honest, just it's I wasn't it's a very complex paper, and it's one of those
Yeah.
papers like the evolution of psilocybin in nature paper. Like again, it's not one of those things you can just summarize in like 3 minutes.
Right. Yes. You
Like
can
the
definitely.
horizontal. So if you type on that little like brief 2 hours of research on that paper and some type in horizontal gene transfer into Google and just download the first three papers, you're talking about like decades of research to understand even
Right.
like maybe this is not something we can explain on a podcast to
Right.
like home
Sure.
mushroom growers.
Well, you did
Like
good. You gave people a
like
taste, though. You know that sometimes that's all we're really doing is giving people a taste or a concept about something. I mean, of course, what we're doing here can't ever equate to an entire thesis or a 400-page book or anything like that. We're just giving people a taste of stuff for
That
sure.
is an interesting point I would like to make. I've noticed that a lot recently. I think we were going to talk about this too. The idea of nomenclature and taxonomy is that one of the things I wish people would remember is that you're a home cube grower. You're not a taxonomist.
Right.
You're not a nomenclaturalist. And we've got these people in the community that just seem to think all of a sudden they're a fucking expert on nomenclature
Right.
and taxonomy, and they just are making these ostentatious like claims that this is the right fucking name, and if you don't fucking change the name on your website, I'm gonna come after you and fucking burn yours. And it's like, dude, when was the last time you like wrote a paper about like nomenclature, or when was the
Right.
last time you like picked up a monograph on that genus, or like I don't know, but we've got these people that have like self-designated themselves like experts, and it just like blows me away. I'm just like, oh my gosh, like, and they're like threatening other people with like if you don't follow this system, you're gonna get in trouble. You mean the same system that last year you said was a different system?
Right. Oh,
And
there's
like,
no
oh,
no short supply of those people, that's for
I
sure.
know they're like
Yeah.
self-appointed experts, and I'm just like, oh my gosh, the audacity, I guess, of some of these people. It's comical to me. I'm
Yeah.
like, oh, I guess they're the expert now.
But I mean, we've seen that countless times. People
Yeah.
designate themselves the police of crosses. People designate themselves the expert of X, Y, and Z because they read one paper, or they're like, but I
Hmm.
took a whole day to figure this out and I understand it now, so now I'm the expert on it. Yeah, there's quite a bit of that in the community, unfortunately. That's why it's just good to talk to people. Talk about stuff. If somebody is still calling it Natalensis and they're not calling it Ocroceo
The
centrata,
N word. Oh,
as long as somebody, yeah, there you go. In
strike.
mycology, it's the new N word. There you go. But as long as
Strike.
people know what they're talking about, it's all good. Sure, there's some instances where these days, I mean, quite honestly, most people don't seem to be caring too much about the real gnats. Most people seem to still really want to talk about the old gnats, the Ocroceo centrata gnats, and that seems to
Hmm.
be a way stronger interest. Maybe that'll change eventually, and it will become more and more important to get that nomenclature right. But for right now, I don't see it causing too many snags, to be perfectly honest. All you got to do is say, "Hey, I see you're selling gnats. Are they the new gnats or the Ocroceo centrata gnats?" And then someone can answer your question, and you can just decide to buy it or not instead of yelling at them like Ed's talking about. Yeah. Yep.
Or they'll be like, "What the hell are you talking about?" I don't know.
But come on, Ed, you're surprised people act like this? I mean, the level
I
of
know.
obsession with naming things in the community is outrageous. People
But
want to
it's
name everything. They want it to be a cross. They want just to name it. I swear to God, some people are getting into crossing for no other reason than it's like, well, I want to be able to name shit all the time. Okay, right,
oh
great,
yeah. Oh, I know people
yeah,
that have lists of names for things
ready
they
to
have not
go.
created yet.
Yep,
Like
Yep.
they've never even touched spores, and they've already got
Yeah.
like 6 or 7 names that they want to name
Yep.
stuff.
Exactly. I've had people who show me a cross and then they give me a list of 7 names and they just want my feedback on it. And I'm just like, I don't know if I care. Plus, what are you on? You're on you just showed me F2 fruit. And you haven't even grown it out. You haven't grown it out past there. I mean, I don't think I'm a little bit like Native Americans, right? I want to see you do your thing for a while. Then I can decide if you're going to be like Little Bear or you're going to be like Dances with Wolves or whatever the fuck, Native American name I'm going to give you. But this coming up with a name and you've not even done the cross yet is definitely not my deal. Yeah, I cannot relate to that desire. It's not my thing. But people like to
Right.
do it. They do like to do it. They want to just, yeah, they got a name. Oh my God. What should I cross? I want to name a fruit starfucker. What should I cross to get that? What I'm thinking, Starry Night, and maybe I know I could use Ed's Ron Jeremy. If I cross Starry Night with Ron Jeremy, with that
There you
starfucker,
go.
is that about right? Do you see what I'm saying? Like, that's the little
Yes.
game. That's the most exciting part of it for them.
Be some
Is that? Yeah. It's all
yeah,
good. That's
something
okay.
that can be construed as a sexual innuendo too.
Sure.
Yeah, that's always good.
There you go.
So-
Yeah. Yeah, but I mean, I
not
get it, but also it's not my deal. It's not. I just like Ed was saying, I like the practice of growing the mushrooms. I like the grind. I like the day to day. Yeah, I like learning the different aspects of it. However, naming is not the most exciting part for me at all. Look at Ed. I already know Ed feels the same way. I don't think he's named a single one of his crosses.
Nah,
It's
I
just
got
this
like
cross
2 or
with that.
3 names.
Yeah.
I'd rather let somebody I just tell other people to name stuff. I don't know why. I feel like sometimes I'm the weird one because I don't feel this compulsion like other people do. Like I feel like I'm the odd one. I'm like, why don't I feel like other? I feel like I'm like going further and further away from like what normal humans do. I don't know why. I think I'm purposely.
What
I'm
do
getting
normal humans do? Tell me.
They sit on TikTok and they talk about politics and they talk about shit that they watched on Netflix.
Yeah.
And like this is what 90% when I meet like a 30-year-old American guy, I met one about 3 weeks ago and he literally had fucking nothing to talk about except for what he had just seen on his IG posts.
Yep.
That was like all he had to talk about. And I was like, dude, you grow mushrooms. He was in Thailand. It's like, like, can we talk about growing mushrooms or something? The dude had nothing to talk about. Dude, young people are so fucking boring. Like I'm like, what is wrong with you? I want to slap them in the face and be like, what the fuck? Do you have any emotions? Oh wait, no, you're on a bunch of SSRIs and antidepressants.
Yeah.
So
now,
no,
have you
don't
watched
have
them
any
though?
emotions.
Have you watched them? Some guy will like start talking to a girl, and if she remotely gives him the time of day, and he gets the sense like she might be interested in him, instead of sitting there and talking and flirting and getting to know her and building chemistry, instead he just asks for her IG, and then leaves. I literally watched it happen before. I'm like,
That's what
wow,
they do here,
bro. You
the
couldn't
Thai
wait
kids.
to run home and text this person that was right in front of your face.
That's the way they do it now.
Okay.
So I heard 2 girls saying that like if somebody talks to them for more than about 3 sentences, they feel like that's weird, that they feel uncomfortable, like with that interaction, that human interaction. They'd rather communicate through texts.
Yeah.
So they can go look on your IG and see, what kind of person you are and what other interests you have instead of asking you. They can go find it out from your Instagram profile.
Go do their little Facebook stalking, and then they think they got you all figured out and go from there. Yeah, man, it's weird. It's weird. I walked into my oldest daughter's room one time. She had 2 friends over, and they're sitting on her bed. They're all on their phones, and I go, hey guys, I got an idea. Why don't you guys actually talk to each other? And I thought I was being hilarious and making a point. And the one friend looks up and says, "We are talking to each other." And I just wanted to cry. I was like, so you guys are all sitting there. They're all on some like Snapchat together. So they're like, we're talking to each other, but then to some other people. And I'm like, y'all don't just want to put those things down and just talk to each other in real life. Nope, they don't want to do that. Yeah, it's sad. And it's definitely going to have an impact on our society. I don't even think we've remotely seen a taste of it yet, of what is about to come. The social anxiety, the weird mild mental health issues that you know are just going to get worse. Anxiety, depression, suicidal ideation, you just, that stuff is just like the most common shit I see in the ER now all the time. Yeah, it's not cool. Not cool. All right. Since we're okay, we're leaving on a positive note. All right, guys, we're giving away some genetics next week. Go to mycogeeky.com. I got a very abbreviated version of the website up finally after sitting on it and never having enough time in the day to get anything up. I was like, I got to start doing this. I want to start giving away some genetics and having some fun. So get signed up. We'll do the first giveaway starting next week. I got a surprise cool guest next week, and then the following week we're gonna have a special announcement. So all sorts of fun, and yeah, we're gonna keep talking about science. When we got science to talk about, we're gonna talk about cultivation. And then when spring hits, of course, I'm gonna be getting out in the woods, so we're gonna probably talk to more, foragers and all that, and yeah, just keep doing it. More integration, more talking about music, all the fun stuff. Just let me know what you guys want to talk about, and I like to be accommodating. You give me a good idea, and I'll pursue it. So. Yeah. All right, dude. Thank you for doing this.
All right, man.
Sorry, I ambushed you with a paper you had not read. It's okay. We'll get it figured out another time, maybe. I still think you did a great job with the gene transfer. Give people some stuff to think about.
Yeah.
So yeah.
Try.
Cool, man. All right, dude. I will talk to you soon. And yeah. Oh, by the way, I got those two. Ed sent me a couple crosses. If anybody is interested in some free crosses, I forget what it is, but it's black cap crossed with, I think maybe one is a Melmac and McKenny. Okay.
Yeah, that's cool. There's like people are getting like white ones out of that. Like on the F1s, the F1 diamonds, there's these albinos popping out from like diamond transfers. Yeah. It's pretty cool stuff.
All right, yeah. Get in, message me, hop in my Discord. We got a channel devoted specifically to Ed's crosses and people who like to work them. So if you guys are interested, you can go check that out. And if I got some left, I'll get them to you guys. All right, man. Talk to you later, Ed. Thanks for joining.
See you guys.
See ya. All right, guys. Here we go. Overlay gone. All right. We did it. We talked to Ed about some science. I learned a few things. Definitely never-ending supply of things we can learn about in the mushroom space. I think for breeding and all this stuff right now, man, I'm gonna tell you right now, the old double cross, the old double swab, not the double cross. Don't do the double cross, guys. But do the double swab. If you're growing a bunch of mushrooms, if you are harvesting within a 24-hour period, 2 different cultigens, swab those suckers. Just swab one and swab the other, back and forth a couple times, get all the spores mixed up in there, and just plate it right away. Just right away while those spores are fresh. You will be surprised how often you are absolutely getting a cross out of the gate, or you're getting a tub where you got some of the parent fruit expressions, and yet you still got something that looks like it might be a cross. Can you prove it? I mean, not really. No, you can't. Does it matter? No, not really. It doesn't. Some of the coolest crosses, some of the most well-known crosses in this community are absolutely come from that crossing methodology. So don't discount it. It puts that game within reach of anybody that's growing more than one cultigen at a time. It's a great first step. If you're looking to go further, Ed has got countless videos on isolating monos using grab and drag. There's other methods you can use as well. It's not hard to get the cross. The hard part is doing the work afterwards. So either you got to do a lot of filial generations, a lot of growing out, or you got to get it to people, get them to work it and all that stuff. So don't be afraid to give that stuff a go. I always feel like you never know. You're just one double swab away from, the next Jack Frost or the next Makilla Gorilla. You just don't know what you're going to get till you go for it. Dave Wombat said that 100 times. Anyway, with that said, it's about that time. We're going to end the stream. I really appreciate everybody, checking in and giving me, give me some feedback on the show, giving me some thoughts on other episodes. I have been getting some more audience advice, ideas, whatever you want to call it. I love it. I want more, really, please, if anybody strikes your fancy, if you know somebody and you're like, man, this guy is so cool, this woman's so amazing, just let me know. I'll be happy to check them out and look into doing a show with them. So, all right, guys, until next week, you guys know what to do. Go grow some mushrooms.